But maybe Gert, I can start.
Yes, absolutely. Yes. Thanks.
Hello, everyone. Welcome to day 2 of our really amazing symposium that Gert Covenberg spearheaded. I mean, it was an amazing meeting yesterday, and I look forward to the meeting today. Today, we want to start with the mini symposium on translational neural engineering for health and wellness. As many of you know, that we have in the age of a multi-scale, multimodal approach to understanding the human brain. Today we have I mean, absolutely an amazing individual who spans the scales of molecules and cells. And he was, I mean, I'm going to introduce him in just a second. I mean, I really admire Rusty because Rusty told me that I have a hope for the future because my brain cells are not completely gone. They can grow again a little bit. So Rusty was the first one to show that. So that's I really admire him for saying that I have a hope. And so let me introduce Rusty Gage. Rusty is Fred, but you could goes with the name Rusty. One day you should tell us why that is the case, Rusty. I have not figured that out yet. And I asked somebody else, a distinguished colleague from your institute, and he didn't know either. So therefore, I should hear from you on that. So Rusty Gage is, I mean, I don't think he needs any introduction. Everybody knows him. He's president of Salk Institute. He's one of our absolutely greatest pioneers in the area of molecular and cellular neuroscience and in other areas related to that, including aging. Rusty got, I mean, just for the sake of record, Rusty got his doctorate degree from Hopkins University. Then he was in Lund University. And I mean, he's, I mean, done some remarkable stuff. I mean, his work, I cannot say it'll take me 4 hours or 5 hours to even briefly outline the work that he has done in the past. He's got incredible number of awards, including the Christopher Reeves Research Medal, the Max Planck Research Prize, member of the National Academy of Sciences. His, I mean, contributions are, I mean, very, very long. But today he's going to talk to us about he's going to talk to us. I'm sorry, I lost your title for just a second. He's going to talk to us about cellular models of human neural aging, Alzheimer's disease, and beyond. I'm looking forward to hearing he's one of the first people to go directly from a stem cell into a neuron. And so therefore we'll hear a lot about his efforts towards doing this and his efforts towards understanding Alzheimer's disease and going beyond that. Rusty, thank you so much for joining us. It's a privilege for us to have you give this keynote lecture. Welcome, and we look forward to hearing you. And for the audience, as always, please post your questions on the chat site. We can have interactive session if you raise your hand. I'm joined here by my very distinguished colleague, Esra Tassali, in co-chairing this session, and we will try our best to manage questions to the best of our ability. So Rusty, please share your screen and start your presentation. There's enough time to go.
Great, thanks very much, Shankar. That's a very kind and generous introduction. I hope you can see my screen.
Not yet. We cannot see your screen vanished. We cannot see it now anymore.
Oh, wow. Interesting. Do I have to start sharing again? Or?
Yeah, I think you should share again. Yeah.
Okay.
Unshare and share, then it might be good.
Mm-hmm. How's that?
Yes, we can see now. You can go into presentation mode. Yep, you're in absolutely the right mode.
All right. Sorry for the delay. Thanks very much for the kind introduction. I'm not going to tell you about my my nickname that I acquired when I was a very small child, other than to say that I'm the third Fred Harrison Gage. So they had to come up with some alternative name, and there was a reason for it. But I'll tell you at another time. Yeah, so today I'm going to talk about 3 different topics. One is talking about aging in human cells and the importance that we all know that a major risk for many diseases is age, and yet we have a very hard time understanding what age really is. And I'll try to show you how we're addressing that dilemma. Also discuss genome instability, which emerged really from these studies on aging, as genome instability is one of the hallmarks of aging. And then finally, if I have time, I'll give an update on our latest work with glia-enriched organoids, and I won't go into that. I'll save that until I get there. So we all know that age is a progressive decline and loss of fitness with time. There's accumulation of stochastic cellular damage that occurs, and it's the largest risk for many diseases like diabetes and neuropsychiatric as well as neurodegenerative diseases. There are, as have been noted many times, a variety of hallmarks of the disease, including, as I said, instability, telomere shortening, epigenetic alterations. And really the way most people do this is they pick one of these and they study them to death. And we have, at the Institute, have been getting experts in each one of these areas and working together on same samples to look at the homeostatic interaction between these different systems. And I hope I'll sample a little bit of that effort with you. So I'll talk a little bit about Alzheimer's disease. And the way most people have studied Alzheimer's disease is to highlight the primary features, like tau tangles, A beta plaques, and looking at either cell models or small organism models, make these human mutations in the cells or small organisms and then study them. The issue, however, is that, how well does this really replicate the disease itself? And since more than 95% of the disease is really sporadic with unknown genetic underpinnings, probably polygenetic, how can we study somatic disease? Well, or sporadic disease. So one way many of you are familiar with is using induced pluripotent cells. So take fibroblasts from human patients and then induce iPS cells, and these cells can be induced further into essentially any other cell of the body, since these are really multipotent or pluripotent stem cells. In addition, one can generate neurons. So we did this initial study where we took patients or individuals, healthy individuals from 1 day old to 80 years old, reprogrammed them to iPS cells, differentiated them to neurons, and looked at RNA-seq to see if we could determine any age-related genes. So here we look at primary fibroblasts. In fact, we can find some age-related genes that are highly significant in age relative to controls. However, when we look at the iPS cells alone, no derivation, just the stem cells alone, we see no differences. And further, when we look at the neurons derived from iPS cells, we don't see any differences between young and old. So we've worked with another approach, which is to take two pioneering factors, NGN2 and ASCII1, in addition to small molecules. We've further elaborated on this through several publications over time, to induce the fibroblasts directly into neurons with the hopes of retaining that differential expression that we saw in the fibroblasts alone. And these pioneering factors are thought to open up chromatin at certain locations, allowing for the direct reprogramming of the cells into neurons. There's a variety of people working on just these factors alone to see how they do this. Is quite remarkable. So on the right-hand side, you see, let me see this, if you can see my arrow. So I'm sort of using this as a pointer. Is that?
Yeah, we can
Yes.
see it.
Yeah, great. Okay, so you can see quite remarkably, these fibroblasts within 21 days are converted into neurons. I still find it almost remarkable that they do this. Now, when we look at differential expression of directly induced cells, we find that we now can find a variety of genes that are age-related. In our first publication on this, we focused on a nuclear pore protein, RAMBP17, which is actually an important beta. That means it's involved in importing proteins, molecules into the nucleus, and it's dramatically decreased with aging. But we also showed that, and when we did a variety of studies showing that this was an age marker, not only in neurons, but also in fibroblasts. And so we're spending a lot of time on looking at ways to activate it. The good news is that when you when you induce an aged fibroblast into an iPS cells, it reverses the deficit that you see in the fibroblasts. So that reprograms the cells. So that means that it's not a genetic mutation, but rather it's a manipulatable mutation. And we're quite excited about that part of it. So since the initial publication, there have been quite a few other studies that have used the vector systems to look at epigenetic, neural stress, heterochromatin, and proteostasis in these aged cells. And overall, they've proven to be a useful model system. And we put these vectors and the subsequent ones on AdGene so that they're freely available to people in the public. So one example of this aged retention is if you look at the fibroblasts that have been, and their direct derivatives to neurons, the INs, if you look at this epigenetic clock that we did with Steve Horvath, you can see that the chronological age compared to the DNA expression, there's higher in the these age match. So they're in their 60s and 70s as per the epigenetic clock, whereas the iPS cells, iPS direct neuroprogenitor cells and neurons are all prenatal. Another example of this is to take the BrainSpan human atlas of the RNA-seq that the Allen Institute acquired and derived a series of what they call genes, neuronal genes, and mapped them at various prenatal time points and postnatal time points. And what you can do then is take your RNA set and throw it into this transcriptome and gives you an indication of how old they are. So if they come from this period, they're prenatal, weeks post-conception, and this is postnatal. And when we look at our data sets, we see that the iPS cells are highly correlated, so the similarity score, they're highly correlated with this early period, and then their lower correlation with as they get older. And that's just the inverse with the IN, so that they're more highly similar to older individuals. Just another example of how, how that is. So we have worked with Doug Glasgow and others at the UCSD ADRC and acquired populations of fibroblasts from well-characterized non-demented controls and sporadic ADs, as well as some familial ADs. But the focus really is on sporadic ADs, our fibroblasts, and then we continue our work. So taking all of these populations and what you see in fibroblast form, Vimentin, so they're early fibroblasts, within 3 weeks, we get a conversion into neurons. We do some quantification showing the NuN, V-glut, and GABA are all equivalent between the controls and AD. So there's not a differential expression of this phenotypes or fates of the cells in these populations. And this also gives you an idea that about 75% of the cells are glutamatergic and about 15% are GABAergic. If you look at this ratio between A beta 42 and 40, interestingly, in the sporadic forms, that doesn't seem to be a marker, but obviously in the AD mutations, that is clearly a good marker. So that's an interesting note. Let's see if I'm doing this right. Let's see. Okay, so yeah. So let's look at the expression data now in between groups. And what we see is that when we look at the fibroblast between controls and ADs, remember now these are age match controls. So whatever age associated differential change is already built into this. And when you compare that with AD, the fibroblasts really don't reveal any substantive things. We're looking at this a little bit deeper, and I would say in the last 6 months, as we dig deeper into this, I think we're beginning to see some differences even in the fibroblast. But when we look at the INs now, we see dramatic changes between the controls and ADs. So there's lots of genes that are different between the Alzheimer's disease and controls. When we look at the transcriptome in general, and we control for sex, age, ApoE, an FDA FAD mutation, we get a nice separation between the controls. Actually, when we look at this group here, look at the controls, we take biopsies from these patients and then follow them. And it turns out Doug has informed us that some of these controls have actually converted to AD subsequently. Now, trying to think what's a good gold standard to compare this to. So we look in the data sets for previously published work from postmortem tissue. This is mostly bulk analysis over the last 15, 20 years. And what we see is there's a very high correlation between the genes that are downregulated in Alzheimer's disease, hippocampus, and temporal cortex and hippocampus, and a good correlation with those that are upregulated as compared to our data set. There's one outlier here that we don't really understand in this parietal lobe that differs. So a bit of confirmation and little more confirmation. There's a single cell study was published by Grubman et al. 2020. And if you do a single cell analysis on this and isolate out of that chunk of tissue that they took and did their single cell sequences, you find the neuronal population here, where they're looking at differentially expressed genes between, again, age, match, and controls. They have 528 genes. If you lay this out and correlate our data sets with neurons, the oligodendrocytes that are here, microgliastrocytes, we only find a really high correlation with neurons, as you might expect. Sorry about that. Now, if we look at genes that are downregulated, we see that an interesting set of classification of genes. I'll focus on this group here. These are GO terms, neural rest targets, synaptic LTP, synaptic transmission. Basically, what we see are a lot of mature neuronal gene sets are downregulated. Neuronal identity is actually decreased in AD. This is a phenotype that we're finding consistency is a loss of maturation in the cells. Synaptic function and maturation are all downregulated. We went back to the cells, made some more INs and did some analysis. We look here at correlation between synapsin and PSD95. We see there's significantly fewer synapses in the AD group. We do morphometric analysis and we find that they have smaller branches, less well developed. And finally, we did some calcium imaging and get a slower, show fewer frequencies of spontaneous network activity in calcium with live imaging. If we look then at the genes that are upregulated, this is also was a surprise. What we find is that there are genes that, well, I guess depending on your focus, we see genes that are associated with cellular stress, apoptosis, and quite remarkably with cancer and stem cell signaling pathways. So we see some of my favorite genes in here, the YAP-TZ signaling pathway, Notch, HIF1 alpha, TGF signaling, cell proliferation, NF-kappa B, inflammatory kinds of signals, and then a bunch of cancer kinds of things are upregulated as well. So what's the connection between this overrepresentation of stem cell cancer signaling and this de-differentiation or loss of neural fate that we're seeing in these neurons? Well, one of the things that's quite clear is if we just focus on genes that are involved in DNA damage and DNA repair, there's some severe dysregulation of these cells. We also see an upregulation of ROS and an increase in DNA contents, as if the cells were trying to go into a cell cycle. We look at that a little bit more carefully. We fact sort the cells for neuronal purification using PSA-NCAM as our marker, and then look at ROS signaling. We certainly see an increase there. And then we look at H2AX as an indicator of DNA damage, and we see an elevation in the amount of nuclear fluorescence intensity in the AD cells. See this in here compared to the others, and another evidence for this increase in DNA accumulation in the PSA-NCAM cells. Regarding cell cycle reentry, we looked at a variety of genes. We'll focus here on cyclin B1 and shows an increase in cyclin B1. Now the question is, do they actually cycle? So we labeled them, and these are mature cells, that we thought were non-dividing, We label a BRDU or EDU in this case for 72 hours and stained, and we see no evidence that the cells actually cycle. They're shifting, but they're not actually going into cycle. So cell fate instability seems to be one phenotype. And so what's happening at the epigenetic level? Can that give us any insights on what's going on? So we used ATAC-seq and looked at controls versus AD, and we see elevated levels of peaks in the AD relative to controls around promoter sites. And they say more globally, there's a greater opening of epigenetic landscape. When we look at where the opening occurs, a significant opening occurs around promoters. And if we look at the Go terms for those open chromatin sites, we see quite remarkably that they're associated with cancer and metabolic and apoptotic genes. So there's a chromatin reorganization that's permitting the expression of genes associated with these effects. This is looking at a motif enrichment, more so you can dig deeper, a little bit deeper into this. And what we see here is LDHA, HK2, important genes in glycolysis, early stages of glycolysis. This is something that we showed before early on and hadn't paid as much attention to it, but it keeps emerging. What's happening quite clearly in AD, it happens a little bit in aging, but it's dramatically enhanced in aging, and that is there's an increase in glycolysis, a decrease in oxidative phosphorylation, and an increase in glycolysis. There's also increases in genes associated with cancer and other chromatin remodeling genes. And this is just, these are differential expression and RNA-seq due to the, we think due to the ATAC-seq. For some of these now, we're overexpressing some of these upregulated genes in wild type cells or in age wild type cells to see which one of them, and then look at some of the phenotypes that we've seen. And we haven't done, we don't report that here, but we're working on that right now. So is this phenotype age dependent? What I mean by that is I've claimed that with iPS cells, we couldn't see any differential expression, but it could be that there's something else going on here that we need to understand. But the advantage is we have these isogenic cells. So what we can do is we can take the same patients, the same 30 some odd patients, and IPS, reprogram the IPS differentiation and then differentiate them into neurons, or we can take them directly into neurons. So now we're comparing the same patients across two different sites, one which retains aging and one doesn't. So we compare the patients. So the value here is that we can use the same this same reporter, or same differentiating vector that we built, the LVUNA, in the fibroblast as well as in the iPS cells. So we have a direct conversion, but all from the reprogrammed cells, and a differentiation directly from the fibroblast. We fact sort the PSA-NCAM population and examine these cells for RNA-seq and compare them one to the other. So if we compare iPS cells, that's at this stage, at this stage, here comparing AD from controls, we really don't see any difference as predicted, because they've been reprogrammed, and we didn't see anything with aging either. However, when we compare the iPS-derived INs at the neural stage between control, we also see essentially no differences between them. So this is all the Go terms that we'd seen before. So there's no significant differential gene expression in iPS-derived neurons from the same cohort that we found these profound differences in the INs. I really don't want to say there's no differences, and I don't also want to say there's no value in the iPS cells. There's just highlighting the fact that the age, I think, is a key feature that you can pick. And I think this may be something to think about if you're looking at sporadic diseases. You may, in the familial disease, may allow you to see defects that are more associated with mutation than we pick up. And we're beginning to look at that as well. So at this point, I'd like to hopefully the questions when you come to this, I can elaborate a little bit more on this de-differentiation that we're talking about. But I think some of the phenotypes that we see are a neuronal loss, a loss of neural functionality, circuitry. There's an epigenetic destabilization, activation of cell death mechanisms, and a partial reentry. Just as a sort of a heads up, we view this, interestingly, as neurons in Alzheimer's disease are undergoing sort of Warburg-like effect, where they are reaching back into a glycolytic program. And perhaps it's because I don't think they're really trying to cycle. It's a standardized oxidative stress on the cell, but it may be that they're needing to resort back to glycolysis to go into the phosphate shunt pathway to get more bulk, to get nucleotides and amino acids to really repair themselves. This story is elaborating. So I would like to say that for our group, what we're thinking more about is that there's a homeostatic, systemic homeostatic state that is normal aging, and all of these different elements of brain function can compensate for each other periodically as you decline. But if there's a second hit, then there is a systemic overstress, and then any one of these disruptions, which were compensating previously, can result in a catastrophic decline, which can be perceived of as cognitive decline in the brain. So that's a conceptual framework that we're building on, not necessarily a conclusion. We should try to understand the way various systems talk to each other to address neurodegeneration and not focus exclusively on one system or another. Okay. So I want to remind you that one of the highlights that we saw here was DNA damage pathways are differentially regulated between the controls and AD. And we have thought about this a lot, and we're not the first to see this dysregulation of DNA damage and repair, but we wanted to get an idea of, when we began to think about it in depth and talk about it with our colleagues in the field that are more expert in the field of DNA damage and repair, we wanted to ask questions about that. So I'll now initiate this sort of second part. So there's actually good evidence suggesting that genome instability is at least part of the problem. So genome instability is directly linked to aging. We see all these various syndromes, Born's, Werner syndrome, Cockney, that are associated with DNA damage and in aging brain. We also see even that there are vulnerable neurons in glia accumulate DNA strand breaks in Alzheimer's disease, some of Leonard Mucke's data. And others have focused on the idea that chronic DNA damage response triggers altered metabolism, proteostasis, downregulation, mitochondrial biogenesis. So the story is set for this being a player. Of course, honestly, this doesn't fit with what I've just been saying. We have to look at each system separately, but we have to look at each system as they integrate with each other, but we have to understand each one as well as we can individually as well. So here's a deeper dive. So what sponsored this or encouraged this study was Dylan Reed in the lab, who had strong experience in DNA damage. And I was saying, Dylan, if there's an increase in DNA damage, where is that damage? And do we know, do we know where DNA damage is focused in the central nervous system? Is it randomly distributed throughout the genome, or are there hotspots? And there was really no good evidence for that. There was some hint that there might be more damage in open chromatin where activity was going on in the promoter region, but that was that turned out to be somewhat controversial. So we developed a technique that we call repair-seq. So we take our directly reprogrammed neurons. In this case, I'll be talking about iPS cells that have been directly reprogrammed to neurons. And in future work, I'll be telling about the INs and Alzheimer's, but I'll just focus on the technique for this point because of time. And actually, we're still in the middle of analyzing data. So what we do is you take the neurons, post-mitotic, we label with EDU for some time, allowing for EDU to incorporate. And I guess you all are familiar with the fact that essentially every cell in the body undergoes as many as 10,000 DNA damaging events every day. So every cell in your body is undergoing thousands of DNA damaging events every day. So we just don't do anything to the cells, just let them incorporate EDU into damaged sites. We do genomic isolation. We do click chemistry with biotinazide, and then fragment the genome, make libraries, pull down the biotin linked DNA strips, and then we do PCR amplification and then do sequencing. And here's an example of what this looks like. This is actually alpha-synuclein gene. And what you see are peaks of repair occurring in the cells. And the peaks are about 500 base pairs wide. And this is just various ways showing how it's replicable. The same sites come up if you do the cells twice, if you do different IPS cells, if you compare IPS cells from ES cells, we get very high correlations in terms of the sites where they are occurring. I should say that if you look at the total amount of DNA repair areas, we find 61,000 sites, but if you look at peaks, there's significantly fewer peaks. And when you look at the peaks, we see that there's an enrichment in the promoters. That's the first piece of information that we can confirm. Not necessarily open chromatin, but in promoter, not open promoters. If we compare the DNA repair with ATAC-seq and histone K27 acetylation, we see that there is some level of correlation, but most hotspots, that is hotspots, not just DNA repair, are not associated with ATAC or with, but are the most heavily repaired sites tend to be associated with them, but it's not a perfect match. But again, when we look at, we do see that 2,500 of the 61,000 DNA repair hotspots are found in observed quadruplexes. So there's an area of vulnerability that's being repaired under normal conditions. And again, when we look at where the impairment's occurring, we do see promoters as being mostly affected. If we look at hotspots and distinct transcription of neural genes, we find that DNA is associated, reads roughly correlates with transcription if we look at all of the sites. But if we take the hotspots, we don't find that correlation with activity. If you look at GO terms, we do remarkably, this is where the hotspots are. They're in neural projection of development, neural development, neural neurons, differentiation, axon. So it looks like the genome is concentrating on repairing damage that's occurring in a regular event in the fate of the cell where the damage is occurring. Now I have to say that we've done this for other cells like fibroblasts and astrocytes, and the picture is not quite as clear, but we do see more glial genes being repaired in glia, but we haven't published that yet. We used a pull down methodology to see what proteins are associated with these hotspots, and they're not necessarily bound directly to it, but in close proximity. And we find 79 enriched proteins. Most of them are histones and RNA binding proteins as well. But 21 of the identified proteins show abundance changes in Alzheimer's disease. So these are genes that have been associated with Alzheimer's disease. So many hotspots, proteins independently were found in proteomic modules associated with late stage Alzheimer's disease. And this is a study done by Dan Geschwin and his group, where they looked at late stage postmortem tissue and looked at proteins that were in modules, modules associated with Alzheimer's disease. And those were very similar to the ones that we found as well, near DNA damage sites. So another interesting point here is that when we looked at the studies that looked at SNPs. So these are small nucleotide variants, single nucleotide variants. It's been reported there's an upregulation of these with time. And so the question we were posed was, what is the correlation, or what's the relationship between these single nucleotide variants and our hotspots? When we look at it, there's essentially no overlap between the two. And we developed or used a methodology for looking at how close our DNA repair is to a relative distance to anything like a single cell nucleotide variant or in evolutionary conserved genes. So this is the sort of standard equation we use for measuring it. And if we compare now the distance, relative distance from these, we find that there's no difference between ATAC-seq, repair, and random. So there's no association of repair. So it's interesting. I'll come back to the analogy that I'll make in a minute, but it's as if the genome is allowing those areas that are being damaged to be damaged, and they're protecting other areas. And one of the areas they're protecting are these areas called evolutionary constraint regions. There's a high correlation between repair in that area and proximity, let's say, to genes that are in that area. We also looked at working with Stephen, we did a comparison to his epigenetic clock. So we looked at the genes associated with aging and used the same sort of strategy of looking at distance. And what we find is that there is a closer proximity to genes that are involved in epigenetic aging. So generally, in sort of in brief, then at the end of the paper, what we did was to damage the DNA exogenously as a challenge to see what would happen. And what happens, depending upon the DNA challenging molecule that you use, you lose peaks that are normally repairing and you pick up peaks in other areas. In other words, and we'll have to elaborate on this, but the hypothesis is that you have a set of areas in a healthy cell which are being repaired on a regular basis. But then when a challenge comes, because of a challenge to a different part of the genome, some of that focused repair will go to another area. But what that means is you lost peaks means that area now becomes an area vulnerable to DNA damage. So we think that this model holds that you have sort of a threshold for repair, things going along fine with DNA repair, and then there's some sort of point of no return where you can't really protect yourself. And part of this clearly is a loss of mitochondrial function. This shifting from mitochondrial ATP-driven energy to glycolysis leaves much less ATP to feed this DNA repair system, which is so crucial for the protection of the genome. So this is the analogy, or this summary. It's like DNA repair in neurons is not random. There are hotspots for DNA repair. The promoters, splice sites, and DNA binding proteins are targets for repair. In neurons, neuronal identity are preferentially repaired. There's a focus on, there's a program of some sort that we don't understand where there's a targeting of these neural identities. Evolutionarily conserved regions are also preserved, and as are biological clocks, which are associated with genome protection as well. The analogy here is that during World War II, when there were bombing raids from Britain to Europe, there was a group of mathematicians in Britain who were trying to figure out why some planes would return and others didn't. So they looked at all the bullet holes in the planes. And one hypothesis was, of course, well, we'll have to reinforce those areas where the bullet holes are. And a very clever guy said, no, that's not what you want to do. The planes that went down were the ones that had damage here. The ones that came back had no damage here. So this is what they ended up reinforcing, and they increased the number of planes that came back. This is the bullet hole misconception. So in the last, do I have a little bit of time left here?
You do, Rusty.
Okay, so let's talk about new methods for analyzing organoids. So many of you are familiar with organoids. They're self-aggregating cluster of cells. You seed them, and under appropriate conditions, they'll aggregate, form a ventricular zone, and then with time, they can migrate out and find a little bit of a cortical space here. The big problem is that over 100 to 300 days, there's a dramatic increase in tunnel. Cells die, or the core of it dies. You get some survival on the outside. So we developed some years ago now a methodology to try to enhance survival. And this is actually a surgery that I developed in 1984, and where we make a lesion in the retrosphenial cortex overlying the colliculus, spiricolliculus, and there's a very rich vascular bed. And we were transplanting superior cervical ganglion in those days. But I thought, well, maybe we could transplant them there, and they get this vascularization from the vascular network. And then what we do is so here's just an example of a GFP positive IPS derived organoid that we implant here. And then you can see that it's clearly, after a period of time, it survives well. Here it is sitting up here in the retrosphenial cortex. We see at 50 days good survival, a little bit of astrocytes in there, but we've developed a new method for enhancing that. And actually the axons from the graft would go out into the host and as far over as the contralateral side. And we published this in 2018, did a lot of electrophysiology showing that they were intact. But one of our concerns was that there are very few astrocytes, and the astrocytes really don't even show up until much later. And as we know, glial cells are essential for brain function. Otherwise, you're looking at organoids, you're looking at primarily a neuronal organoid, not really a brain organoid. So we developed a protocol to enrich for astroglia. And here's what we see. So here's a forebrain auricle. Without our program, we see no GFAP in this organoid, even at 8 weeks, whereas in the aged, so in the glial enriched organoid, we find lots of GFAP. This is even in vitro. We find them, and this is just a quantification of that. When we transplant them, so in vitro, you get this massive amount of astrocytes increasing in the organoid, but they're distributed in weird ways, and they're overlapping. When we graft them, they now become much more distributed, and the actual number decreases, but they're tiled much more like they are in the CNS than they are in the in vitro setting. One of the things that we noticed, aquaporin, which is a good marker for vascularization, we see that the astrocytes engulf these vascular, and I won't go too much in detail on this. We're still working on it. It's actually under submission. But aquaporin associated with astrocytes, and obviously in vitro, we have no blood vessels, so there's no association. I can also say that we've identified specific types of astrocytes, and there are astrocytes in humans that are unique to human that don't exist in other species that are being revealed in this methodology as well. So one of the things we can do is we can put a glass cover slip over the top of the organoid, and then we can use 2-photon imaging. And what we do is we'll inject a Lexaflora 594 dextran into the eye, retroorbital injection, let that flow, and then we look with a live 2-photon microscopy. And here you can see in live imaging, the dextran, this is inside the organoid with 2-photon. You can see that there's the surrounding cells. And now we have rich glial encasement of the glial with very little leakage of dextran into the host. If we do this now take ultrastructure images of the of the vascular net, we find this, well-described vascular unit inside the human organoid. Green is astrocyte. Remarkably, orange, or whatever this color is parasites. Pink, purple are endothelial cells, and this is the extracellular matrix there. So that comprises what we traditionally think of as the vascular unit. These are tight junctions here with the endothelial cells required for that blood-brain barrier and the flow of dextran without any leakage that we saw. We did three-dimensional reconstruction of these images, and you can see that the astrocyte is now fully engulfing that vascular unit, suggesting that these are recapitulating vascularization in this human host organism. So in summary of this part, we have a rapid derivation of functional astrocytes in glial-enriched organoids. Astrocytes form spatial territories. They're patterned with astrocytic infeed in vivo and transplanted, blood-brain barrier formations within the transplant. And we've done RNA-seq on this that I'm not going to show you right now at single cell, and identifies different types of astrocytes, some of which are unique to humans. So with that, I'll stop and thank all my fabulous colleagues, postdocs, and collaborators for their work in the INAD, the group helping or driving this repair-seq work, and two fabulous postdocs that have been working on the astrocyte organoids now for 3 years. And hats off to our cores at the Salk and the UCSD Alzheimer's Disease Research Center, which have been so tremendously helpful in our work. And with that, I'll thank you very much and answer any questions. Well, I'll try to answer questions.
Thank you, Rusty. I mean, outstanding, amazing work. Thank you so much. I'm going to, before I open up the floor to everyone for questions, I want to take the prerogative as chairing the session and ask you a few questions. Thank you very much. Rusty, as you know, I mean, I want to lead off with saying
should I stop sharing
yeah, I think that'll be good. You can stop sharing. That'll be good.
Okay.
Fantastic. This will also be good. Please raise your hand if you want to ask questions so I can call upon you to ask questions. We'll monitor. I mean, there are too many actually, but we'll try our best to monitor as we go along. So Rusty, as you well know that, in iPSC-derived neurons, we had shown the dedifferentiation endotype and other related, initiation of aborted cell cycle and loss of synaptic function as endotype. So my question to you is that we didn't work on late onset sporadic AD. But it looks like all the endotypes that you are seeing in INs are captured in iPSC-derived neurons in the presenilin-1 mutation ADs. But the question for you is that when we published this, we showed that REST and REST-related genes associated with synaptic function are differentially affected in iPSC-derived, FAD neurons. And there's loss of synaptic function associated with that. So do you see something very similar in your, IN, sporadic AD INs?
Yeah, absolutely. REST and REST-related molecules came up with one of the top categories are Go terms. And I guess it was Brad Hyman and their group that showed that some time ago with REST. And we used to work a lot on REST. So that I think of that more as a part of this idea of neural identity. Yeah. I think it's one piece of a larger process that the cells have more open chromatin that's allowing them to open areas and this loss of oxidative phosphorylation moving into glycolysis. All of that is encompassed with this. One thing I haven't talked very much about is that we do see elevated levels of senescence in the compared to controls. And I think we think of senescence really as being part of this Warburg effect. So instead of turning into a cancer cell,
Yeah.
cell actually senesces and ultimately die, as a protection against dying is to senesce.
Yeah, exactly. We published exactly the same thing in terms of safety mechanism the cell is trying to deploy. So related to that is a question that between your iPSC derived neurons and IN derived neurons in the same, same individual, cells, fibroblast cells. Did you do electrophysiology to look at the difference between the different electrical potentials and the types of neurons in these two cell types and how they are different?
You mean between controls and AD?
Between INs and iPSC derived neurons in terms of looking at a comparison, because you showed the aging effect. I'm wondering if there is a consequence to electrophysiology in these two neurons.
In the aging in particular.
INs and iPSCs, from the same origin.
Yeah, so the IPS protocol has a lot of astrocytes in it, and 15-20%, and the astrocytes really promote neural differentiation connectivity more so. So you have to add astrocytes to the INs to get the equivalent. Otherwise, they don't have much connectivity. And trying to get that right ratio so you're making a fair comparison is actually pretty hard. So we're still working on that, and we have a protocol now where we take the IPS cells and the IN cells and co-culture them with a basal level of neuronal cultures from, say, mouse or something like that, so that everybody sees the same, environment that they're in. And we're doing those comparisons now, because it's just, to make that comparison honestly has been more difficult than I thought.
Yeah, thank you. Because that'll be very interesting and exciting if you can get electrophysiology from these two types. And
well, we have two postdocs working diligently on that,
yeah.
right now, and that's their goal in life is to make the head-up comparison.
The one other question, I really enjoyed very much listening about the DNA repair and DNA damage and repair gene story. I was going to ask you if you saw any correlation of these regions. I know you showed the chromatin, the histone connection, but do you see preferential correlation with the DNA methylation regions with these changes, alterations in your DNA repair hotspot, genes that you're finding?
Yeah, the closest we've come to that, to doing that in a global sense, is actually, we're sort of cheating a little bit. We went and targeted specifically the genes that Horvath has identified as being age-associated methylation sites in the genome. And there's no question that our hotspots are very closely linked to. I'm not saying they're on, but they're closely linked to, using our similarity score. So yes, I would say probably not to all methylation sites, but certainly to these. And when we look at HK327 acetylation, it's reasonable. It's not any kind of perfect thing. And I've really went into this thinking that there would be a much greater association with activity than there is, and that open chromatin plus RNA-seq, we did GROW-seq, we did all sort of things, thinking for sure that this was going to be, the vulnerability was activity. Just isn't. It's not very impressively associated with that, but it does go to promoter sites, but even in promoter sites that are not particularly active. So they're actively working on protecting sites associated with identity.
Yeah.
At a philosophical level, what more important could there be is protect the DNA of the fate of the cell that you're in, protecting your fate. I like to think about it. And that's,
yeah, no, that's teleological, very interesting. So therefore, I have more questions, but let me get others into the play, and then if the time permits, I can ask you. Ben, you had your hands up.
Oh, thank you. Very interesting, and I agree. At the DNA repair, I thought was really fascinating. I'm coming from circadian rhythms, and thinking about things we see in aging. You see this deconsolidation of circadian rhythms, and as you mentioned, the circadian rhythms actually play an important role in acetylation and deacetylation to make these sites available. I wonder if you see a connection between those two. I'd just be curious to hear your thoughts in terms of, for example, if it's not activity, is there some sort of circadian coordination required to make the right sites available for repair? And as that fails, that leads to sort of aging deficits.
I think that's a great question. We have been talking with Sachin Panda here at the Institute, and we're trying to get an in vitro model in the INs to have a sort of circadian-like activity in it. And that those electrophysiologists are working with him, but also with our multi-electrode arrays to see if we can find up some phenotype. So any ideas you have for being able to capture the circadian phenotype, I would love to see whether or not if we do get something that's reasonable in young, healthy cells, can we then compare it to controls? But that'd be lovely. So any ideas that you have, please share them.
Yeah.
Either now or in the email sometime.
I won't waste everybody's time, but yeah, I might send you an email. That's very cool. Thank you.
Great.
Other questions for Rusty? I don't see any other hands up, and I don't see anything in the chat. So I'm going to continue asking you questions.
I actually wanted to
I'm
ask.
sorry, Esra. So I missed you. No, please go ahead.
I'm coming from a clinical perspective. I wanted to see, first of all, fascinating talk. Thank you so much. I learned so much, and I have to digest all of that. But from a clinical perspective, at the beginning of your talk, you mentioned how aging is related to type 2 diabetes. And you also mentioned Alzheimer's, which is a very important problem for our public health. So I was wondering, where do you see the translation of this sort of the electrophysiological modeling work? Where do you think we can literally help the patient at the bedside in the short term or in more of a longer term?
Yeah, so one of the things that, one way to look at it, just in a global way, is
Right.
we are really looking for targets that are associated with aging. So we're very excited about this RAMBP17, because that is really an aging marker.
Right.
It's not exacerbated in AD, but if in fact the story is that age is a requirement or the highest risk for AD, can we stabilize the chromatin in us, in the aging healthy population, so that we become less vulnerable to that second hit, which leads into catastrophic disease. So that would be one way, is looking for age-related targets that we could interact with therapeutically. And the other is obviously this. I'll give you a general sense that we have identified some very specific targets that are involved in this shift that's occurring from oxidative phosphorylation to glycolysis. This is using mitochondrial ATP versus glycolytic generated ATP. We think this is a big deal, and there are specific molecules that are shifting, and they're the same molecules shifting in AD that are shifting in cancer. So we actually have now a variety of cancer drugs that are, well studied in trials, safe, that are addressing this particular issue in cancer, that we are trying in our model systems to see whether or not we can shift this energy metabolism back to an oxidative state. I really do understand something about the algorithm the cell is using to shift from oxidative phosphorylation to glycolysis. It makes sense. This is an evolutionarily conserved attempt to stay alive. And yet, it's like anything, too much of a good thing is not good for you. So that, and the analogous area, and I should say, so that's one area that we're working desperately on. The other areas, there's clearly an increase in inflammation. I didn't spend a lot of time on that, but there's clearly an increase in inflammation. And we've identified several kinases that we're particularly interested in that are uniquely upregulated in AD. And so to answer your question, we're interested in sporadic AD since that's the big elephant in the room when it comes to Alzheimer's disease. And I'm certainly not disagreeing that tauopathies and amyloid are important, and that you can target those in a familial form. But I'd like to spend a little more focused time in finding what are the issues associated with sporadic that we could also attach. So our attempt here is really not to downplay the importance of familial, but rather to say, what are there unique features of sporadic that we could target as well?
Right. Thank you. Great.
Yeah, thanks, Rusty. I mean, Esther, I would like to make a comment on your question, but before that, I have another question for Rusty, which may be related so tangentially. You talked about oxidative stress and RANBP17 and the role. Do you know the mechanistic connection between these two and how stress induces damage or alteration in RANBP17 expression? I mean, or is there not, that's not clear yet?
So it's not a damage of expression. It's a, so what happens is there's a downregulation. So we're mapping out the promoter region of RANBP17, trying to figure out how it's dysregulated, but it is the expression that's dysregulated. It's not a genomic event. So as I said, when you make iPS cells out of the fibroblast, that repairs RANBP17. We've made an assay system where we color the genes associated with nuclear import and red for nuclear export. And what we see in fibroblasts and in aged fibroblasts and in control and aged AD cells, neurons, there's a dysregulation. They're leaky. The nuclear pore is leaky in aging. And that leakiness, in my mind, I associate that more with DNA instability. That's allowing stuff to get into the nucleus that shouldn't be there. And, Shankar, this is why I think it's so important we look at all these systems simultaneously, because an inflammation is going to have an impact on oxidative phosphorylation and DNA repair overall dysregulated with mitochondrial deficits. So I think it's really a system-wide analysis that needs to be done and not just focusing on one or the other.
All right.
That's a little bit of hand waving, but
No. I think you're absolutely right on the ball. I mean,
it would be actually interesting to know if one can get a handle of it to see if that leaking is a precipitating factor, sort of opening the path for inflammation. And I'm sure it's perpetuating too, but I think it's very, very interesting. Yeah. Could this be a biomarker that one can measure, say, in clinic setting in blood or?
Yeah, no, I'll tell you a little story that's in other cells, not neurons, that helps to link it for me. It turns out that Jan Karlseder in our institute that works on telomere shortening, he's shown that some of the proteins that bind to the end of telomeres decrease with aging, but those proteins that don't bind, they actually they target mitochondria. They get in mitochondria, and then the mitochondria become dysfunctional and extrude their DNA. The DNA from the mitochondria. That DNA is seen by the cell, by the gas stings environment, the inflammation environment, as foreign, and sets up an inflammation. So here you've linked three systems together: telomeres, mitochondria, and inflammation. I'm not calling that causal in any event, but it just shows you how these systems are so interrelated to each other.
Right.
So Esther, in response to your question, to make a quick comment, since there are no other questions for Rusty at the moment. We had also shown that in familial AD, the same endotype mechanisms that Rusty pointed out in is, sporadic AD are seen, which is dedifferentiation and synaptic loss and cell cycle reentry or potential reentry, senescence. So what we had done is taken those endotypes towards a screening mechanism for targets therapeutics. And there's a paper next week, it's coming out in Alzheimer's and dementia, which shows how the current drugs that were focused very heavily on the amyloid hypothesis, but they don't fix everything. They fix them. In fact, the Eli Lilly drug, which is very famous, went on for 15 years, semaglutide and the other stats. They all showed, loss of amyloids, loss of, dissolution of amyloids, but they didn't restore synaptic function. And so what that tells you is that it is more than just amyloid hypothesis. And in fact, Wall Street Journal had said, published that amyloid hypothesis is dead because of all these heavy clinical trials that have gone on without any success. But now that we have the arsenal of endotypes that we can look at, is a very good screening mechanism. Our paper shows comparison between many drugs, which show how the endotypes together say that what is fixed and what is not fixed in these drugs. So therefore, it's a new way of drug screening for translational effect or clinical setting that you talked about in terms of designing new drugs. And so this should be out in the next weeks in A&E, and so you should look for that. Anyway,
Great.
I just wanted to mention to you.
Looking forward to it. Congratulations on the paper.
Yeah, thank you very much. And I think Gert wants to say something before we take a break. And Gert, please.
Yes, Rusty, thanks for a great talk. This is wonderful. So in the theme of the workshop, right? So you're presenting how these genes can regulate this disease, right? And how there's devastating effects for Alzheimer's and other neurodegenerative diseases. So to what extent does activity affect all this, right? So, and this workshop also embraces like electroceuticals, means for interacting, right, with the nervous system at many levels, right? So at what level can you see the interconnection between, say, activity, neural activity, just electrically driven activity, say, and all those different pathways that you're modeling here?
Yeah, I think in general, you do see an association with activity, but not necessarily the hotspots. It's just general low-level repair around includes that. So the correlation's low, but if we do it with hotspots alone, there's not any correlation. So then what we did was to take KCL. So that's what's normally used in a tissue culture, not normal, but is used in a tissue culture setting to elevate action potentials and get activity going. We saw no change. Maybe we didn't treat long enough. You could argue all kinds of things, but if it was a real robust, we used sort of standard procedures, standard concentrations, could see increased activity in the cells, but we didn't see any increase. So I'm not saying that activity is not important, just this one area of repair we're talking about. It's not AD or anything like that, but I think, the other part, these are all acute measurements that we're doing here. So one could argue that low levels, one area that I also work in is in the dopaminergic neurons. They are extremely active and spontaneously active. And they really rely on a lot of oxidative phosphorylation to keep that activity going. So they have high amounts of mitochondria and a lot of activity. And so that's a chronic throughout their life. They're just firing like crazy. So I think those cells, the theory is that those cells, because of their high level activity, they're highly branched, going to septum, accumbens, frontal cortex, everywhere. They're branches, and they're not that big. The neurons are pretty small. But I think that's an extreme case where activity probably is playing a big role in vulnerability, not causing disease, but vulnerability.
Fantastic. Thank you, Rusty. Amazing talk. And as always, your insights into all these things are profound. And thank you for such a wonderful presentation. We appreciate it.
Thanks very much. It was wonderful being here, and thanks for the questions. And I wish you all the amazing rest of your meeting. Nice to meet you.
Great. Thank you very much. And we are going to take a very short break. We'll be back it's 9:13. We'll be back at 9:21 to start off Ben Smarr's talk. I'll introduce Ben, and my colleague, Dr. Tasali, will chair the session. I'll just introduce Ben and leave you there at that point in time. So look forward to seeing you all back in 8 minutes bio break.
Thank you. And so the breakout rooms are now open. So those who would like to check out perhaps an early version of demos or try the demos, so please, you can go ahead and try the breakout rooms. If you have trouble accessing the breakout rooms, make sure you have the latest version of Zoom installed, which is at least 5.3. Otherwise, you may not just actually enter the breakout rooms. And so looking forward to seeing you soon. Thank you.
Hello, Ben. You want to share your presentation so we can just, make sure that everything works?
Sounds good to me. How's that?
Sounds good. Absolutely wonderful.
Do you want me to go back?
See it. We can see it well. It's in a correct full screen mode. In good shape.
Cool.
And you even have your laser pointer ready.
I'm laser pointer equipped.
Excellent.
That was a hard act to follow.
Rusty gives great talks. So I'm sure, Ben, you'll do great.
That was really interesting. I was Googling while we were doing some more questions, and there is definitely interaction between circadian clock genes and the nuclear pore mechanisms that he was talking about.
Yeah, no question. Yeah, there's also a paper published previously on the same topic of connection between nuclear pore factors and circadian rhythm. But it has to go, I think, going back to it, has to do with the chromatin dynamics as well, which we don't understand in detail as to how the chromatin dynamics in turn is altered in a rhythmic manner. Probably multiple rhythms associated with chromatin dynamics.
Yeah, no, I assume so. It's really interesting to come at it from those two different ends, where you know that the circadian disruption makes that kind of neuronal health harder, leads to things like dementia, and to see that there are these sort of substrates within the cell that are clearly affected by that mechanism. And he's right. The right experiment to tie them together is a challenge.
Right.
But that was really exciting to see all those extra pieces.
It'll be absolutely wonderful to look at that. I mean, I was so hoping somebody would do a high-seq experiment with your different rhythms so that we can try to really study for the same cell and chromatin topology changes overall. I mean, in the nucleus, it's a function of time.
Yeah, no, it's a challenging study, but I totally agree. I think it would be fascinating.
I mean, I think that knowing the circadian implications on health, one would also hypothesize that the repair pathways are happening in a circadian fashion.
Yeah.
Right? Not all the time, but
yeah,
oscillates, which makes sense physiologically.
Yeah, in fact, Ben is going to tell us not just only about circadian, but also ultradian rhythms, which are going to be very interesting and important for us to look at. So I think we are in the right time to begin again. We are 9:24. We are about a minute or two later than we promised. So I want to get started off. Ezra, I'll turn it over to you as soon as I introduce Ben. Ben is, we are really fortunate and privileged to have Dr. Ben Smarr join us to give this next talk. All of us know that, most of us are familiar with the notion of circadian rhythm, day-night rhythm. If we are not erudite like Ben is in this area, we know that there is a difference between light, day and night, and various cycles in the body, and we are quite familiar with it. But when I first saw, Ben's presentation, I learned that it's not just simply circadian rhythm in a bimodal manner or in a discrete modal manner, but there's also something called ultradian rhythm and infradian rhythm. So that you have multiple rhythms happening at different time scales. So Ben really brings a breadth of knowledge in terms of building across the multiple time scales, rhythms in our physiology, and how these rhythms impact our daily physiological function and long-term physiological function. So this is a phenomenal topic. Ben is one of the pioneers in this area. We are fortunate to recruit him from Berkeley. He was, postdoc in Berkeley prior to coming to UCSD. And so we are very fortunate to have him. Ben has also played an absolutely key role recently in the COVID-19 pandemic assessment, and he became one of the technical lead for this company called TempRedict, that a global collaboration that combines physiological data, symptoms report, diagnostic testing, and he wants to build data models. So he's really a true polymath who brings in a lot of intuitive innovation, methodology, and outcomes at the same time. So I really welcome Ben. And Ben, take it away, and we look forward to your presentation.
Well, thank you. That was a really nice introduction. And I hopefully won't disappoint. I'm not going to talk about TempRedict very much. It's a DoD-funded study led by clinicians at UCSF and then a technical team here for which I'm the lead. But I think I talk about COVID too much, so I'm going to try to talk about some other examples of behavior modulation, and I'll just touch on COVID briefly. But from a neuroengineering perspective, I wanted to start and say, we know that our brain affects our behavior, obviously, and vice versa. And so there is some open area, I think, in terms of trying to find ways to understand technologies that allow us cheaply and at scale to understand the neural state of a large population. And it's hard to just measure somebody's brain directly. And so that's where proxy measures become very useful. The first question I want to just lay out is, if I'm going to be talking a lot about wearable devices, what does that have to do with neurons? We just heard a very mechanistic talk. This is going to be more sort of phenomenological. And I see the two as intrinsically connected. And Shankar, I think you did a nice introduction of that for me, so thank you. But as we think about what is it the brain is doing, it's doing a lot of regulation of output. So it's output behavior, output of hormones, and those outputs are rhythmic. They're not tonic. You're not always hungry. You're hungry every 2 or 3 hours. Is one of these ultradian rhythms. Hormones pulse along with these ultradian rhythms. And so not only is the output of the brain rhythmic, meaning our behaviors have a rhythmic quality to them, and our physiology, but those behaviors in that physiology then feed back to our brain. And so as people do more and more in vivo imaging of time series within the brain, they find that more and more regions in the brain show not only circadian rhythms, daily rhythms, but these ultradian rhythms, so every few hours change pulsatility, and then longer rhythms, infradian rhythms. So ultradian is ultradian, faster than a day. Infradian is infradian, so longer than a day. And this includes ovulation or seasonality. One could argue the life cycle. So all of these rhythms manifest in the brain. They manifest pretty globally in the brain. And so when you're thinking about what is a neuron doing in terms of cell repair, interacting with glia, this kind of thing, the time, the phase of those oscillations has a big modulating effect on what is that neuron able to do and what are the outputs going to be. And so you're changing the control surface in this rhythmic way. So just to show a short example of that, this is an older paper, but it's looking at luteinizing hormone, which is part of the reproductive axis. And here you're looking at neuronal spikes, and you see that they have, in this case, roughly a circoral rhythm. So about once every hour, there's this big burst of spiking. And then in the blood, you can see that that agrees with this ultradian rhythm now, again, roughly in this case, a little more than an hour of luteinizing hormone. And luteinizing hormone then goes on to stimulate release of things like estrogens or progesterone from the ovaries in a woman. And those things then affect your heart rate and your temperature. And so wearable devices, by picking up those proxy effects, changes of temperature, heart rate, activity, blood sugar, they're really giving us a window into the rhythmic cadence that the brain is trying to establish. And to the extent that you can help people be aware of that by giving them new dashboards to look at rhythmic qualities of these data, they can then exert behavioral control to try to stabilize those rhythms and avoid the kinds of things we talked about in Rusty's talk of circadian disruption across life, raising risks for things like diabetes, dementia, heart disease, infertility, pretty much anything you want to name. How do you help your brain keep the beat, basically? So a very simple example from a fairly high level of how we use these kinds of data to reveal new things comes in women's health. So I'm going to show an example from pregnancy. And I want to start again with just a question of why is this new? And fundamentally, it's just because the tech hasn't been about. So, wearable devices are very new. The ability to do computation of signal processing over very large data sets is still evolving. The ability to miniaturize these things is still evolving. And so it hasn't really crept into standard medical training, of course, because we're still inventing it. But what that means is, again, there's a big opportunity space for understanding not just structurally what is a person when they're healthy or unhealthy, but what are the dynamics that help us understand that context and help us watch those changes unfold. And that's really the frontier that I enjoy. So this was work that I had done in rodents a few years ago. And I was looking at circadian disruption during pregnancy, which leads to autism-like phenotypes, it turns out. And I was trying to figure out when I should do interventions during these pregnancies. And I asked the vets, how do you know that a mouse or a rat is pregnant? And they said, oh, that's really easy. We solved that a long time ago. When they give birth, then you know that they were pregnant. And I said, okay, that's, that really doesn't help me get ahead of the thing. But I had these little temperature implants that I could use to track their circadian rhythms. And I noticed that there were these temperature rhythms that we could pull out. So here what we're looking at now is the 4-day estrus cycle of the mouse in this case. So that's like the 28-day menstrual cycle for a human. It's ovulatory rhythm. The animals get hotter as they ovulate. And then here we see the onset of pregnancy. There's the spike of temperature. And then there's very different, here we have within the individual where their baseline rhythms are. And then you see that that rhythm structure changes very obviously once they're pregnant. You can see when they're going to give birth. And if you just randomly measure temperature once a day, say, you wouldn't see these patterns. But because it's a continuous measurement, all of a sudden comparing the individual to themselves over time, the pattern becomes pretty obvious, actually. We've recapitulated this in humans, so we'll put this up on medRxiv and it's in review now, but you can watch human menstrual cycles, and you can see the onset of pregnancy already just as a very rough first hash. We developed methods that reliably tell us somebody's pregnant within 4 or 5 days from conception, whereas the alternative is after a couple of weeks. If you think about going and buying a test, you can go and buy a test and then you have to worry about, is it accurate? And so there's rapid gains to be made in this field is the point. And the reason you can see these changes once again is that the rhythms are coming from these changes of hormones. So as the physiology changes, as the neuroendocrine regulation changes, that changes the surface that you're measuring with these wearable devices. So given that there are these big gains to be had, there's this question of why aren't there better health AIs out there? Why can't your wearable devices already offer you a lot of products about, are you aging well? Is your diabetes well managed? Are you pregnant? And fundamentally, as far as I can tell, it just comes down to the fact that people are very diverse. And we've never had a good lens on physiological diversity. So that's another thing that these time series allow. So here we're looking at just the comparison of average and range of temperatures across 50 individuals. And you can see that they differ from each other. Everybody is not 98.6, and these ranges are very large. Most of that comes from circadian rhythms. And so again, most people are not 98.6 at any given moment either. So they differ from each other, they differ from themselves over time. Heart rate is the same story. People have a very wide range in the population. So any linear threshold, like getting your forehead measured when you get onto a bus to see if you have COVID, it's just not very effective. And the reason is that comparing people at random time points to a population average, you wash out all the precision that you gain by comparing an individual to themselves over time. People also, of course, as I've said, have multiple scales of rhythm. Shankar introduced that really nicely again. So here we're looking at an average in the North and South Hemisphere of about 10,000 people each and looking at their daily resting heart rate. And here's New Year's is a big spike, so you can see the behavior, of course, is also affecting these rhythms. And each of these spikes, there's 52 of them, those are weeks. Weeks is not a biological rhythm as far as we can tell. But again, people's behavioral choice about what do they do on the weekends ends up impacting and superimposing on these rhythms. And so again, if you know where somebody is, how weekly their rhythms are, you can contextualize the measurements much better. And this is part of how we were developing detections for sicknesses. Not just is your temperature more than 98.6, but is the rhythm being disrupted compared to your normal pattern? So here, this was the first patient that we ever got that had COVID. And this is all I'm going to say about COVID is this one slide. But here we're looking at 20 weeks history of this person's body temperature. This faint blue line is minute by minute temperature, and then the thicker blue line is a rolling average to sort of summarize that across days. And then we have heart rate, same story, heart rate variability. And in red, what I've highlighted here is there were 3 days here where the individual had a fever, and this was back in March of 2020 already, so an early case being reported relative to the pandemic taking off. And so this is highlighted 3 days before that, 3 days of fever, 3 days after. Now, this individual never felt sick. I spent a fair amount of time talking to them. And instead what they said was, I was looking at my wearable device, in this case it was an Oura ring, and I noticed that my temperature was up, and I knew that there was this virus going around. So I went and I got treated, got tested, and sure enough, it was positive. So I guess that was COVID. Now, those of you looking at these data will notice, A, they bounce around a lot, but B, especially this individual, when they have a fever, you can see temperature is elevated, heart rate is elevated, heart rate variability is depressed. But you can also see that that happens over and over in their history. And in this case, that's because this person is an extreme athlete, they're an endurance runner. And so every week they're going through these training regimens. And so this raised the hope that we could definitely see COVID from wearable devices, which was not obvious in 2020. But it also raised this question of, can we tell the difference in shape and form? Can we train algorithms to recognize the difference from things that are self-imposed, like running a marathon? So one way that, and short answer was yes, and we ended up generating algorithms, running trials, roughly 90% accurate predictions 2 or 3 days before people start reporting symptoms, so that's extremely promising. But one of the other things we were thinking about was, how do you give that back to the individual? How do you help them think about, should I get a test? Should I self-isolate? That sort of thing. And so these time series are very data rich, but they're not easy to digest right away. And so there's this question of, how do you dashboard things that helps a clinician or an individual make sense of the rhythmic information that we're pulling out of this? So this is one example. It was an early example from COVID. The white dots are time points comparing heart rate variability from this 20-week history. And then the colored dots are the things that were within this red range. And the color is proportionate to this individual's temperature at that time. And so what you can see is their baseline is down in this very dense area of white. There's this blue superimposed on it, and that's where they spend most of their time physiologically. And then there's this much fainter white excursion area, which is when they're really stressed out. And in red, what you see is this progression into fever. And then as the dots get smaller, they're getting closer to the present. So you see there's this recovery population down here. And one thing to note is, not only does this let somebody, if you make a movie out of this, you can watch these dots evolve over time. You can see the temperature transitioning. You can see the emergence of the fever. But you can also compare for recovery, that is. And so we can say, okay, well, these small dots are where this person settled after the COVID stopped, or after the fever stopped, rather. And it's roughly within the baseline area, but it's not the same. Right? The dark blue is left shifted from the light blue, which is the before is left shifted from the recovery. And so using approaches like this, we've also been able to monitor things like long COVID, where some individuals show much greater shifts within this landscape that last sometimes for many months. And so in this way, not only can we pull that out of the data and build classifiers, but we can help the individual make decisions about their physiology by making it more observable to them. Now, given that we can't build AIs because of all of this complexity easily, the way that we're starting to learn how to make labels is by capturing individual experiences. So that COVID was one example. And I want to show you an example where we, rather than just say we measured a lot of points and therefore caught the fever points, I want to show an example of where having that continuity of data really allows us to develop better analyses for capturing physiological dynamic changes. So this was, and I don't think this is a surprise to a lot of people, but some of you know this anyway. My dad had to get a colectomy at some point, suffering Crohn's disease. He doesn't mind me talking about this, just to make sure you know that. And of course, being a loving son, I said, Dad, you're going to go through surgery. I'd really love to put wearable devices on you, because that would be really cool to capture that. And being my dad, he said, do what you need to do. So this is Armin Garabands, who was a graduate student of Todd Coleman, who was here at the time in bioengineering, both doing extraordinary signal processing work. And Armin's thesis was this little black bag, which was a gastric monitor. So we could put it on like an EMG, and he had developed some extremely sophisticated noise cleaning algorithms to get through the abdominal muscles and look at the gastric contractions underneath. And so since he'd had a colectomy, we were able to observe how was he beforehand, very spastic. How was he after the surgery? He recovered very well. And right now, I'm just going to focus on how he was in the hospital. So we measured continuously, and then in this case, transformed the gastric contraction data, these linear time series, into a frequency space using a wavelength. And so what we're looking at here is periodicity, and this fuzzy red band at the top is the 24-hour periodicity. So daily rhythms are there all the time, but they're not super strong in the hospital. This is 48 hours after coming out of surgery. So he's in the hospital bed, he's not moving around very much. And when you come out of surgery, there's this period of postoperative ileus where the intestinal, your tube basically from one end to the other, is not functioning yet. It's not peristaltic, it's not organized, and so it's not safe to feed you. It's not safe to let you leave the hospital. And the longer that postoperative ileus lasts, the higher the rate of complications are, the more likelihood of complications. And of course, the more burden on the hospital as you fill a bed instead of recover. The standard for measuring postoperative ileus is the nurse comes in and they ask you whether you've passed gas every couple of hours. So this is obviously both labor intensive and not super precise. What we were excited to see in this just N of 1 was that as he's recovering, there's a lot of broad spectrum activity in the gastric system, so not well organized. But then there's this transition to sort of a focal point of ultradian rhythm power, where he then passed gas, had a bowel movement, subjectively felt much better. And you can see basically it goes through a state transition. All of a sudden now things are much more akin to circadian power. They start to focus around this band of 3 to 4 hours, which is where they then stabilize over healing. And so you can monitor the physiology to help make these informed decisions is the hypothesis. Of course, how this differs for different individuals remains to be explored. Again, since this was a question of not just how do you do the math, but how do you also inform the clinicians and the patient, I built a little dashboard for this as well. So this is looking at the colectomy and looking at time of day, looking at power and stability of frequencies across time for that each moment of the day. And hopefully this is not too shocking, but I thought this made a nice dashboard in the context of taking colon surgery and feeling good about it afterwards. So here before the surgery, you can see there's a lot of red on the outside, which in this case is unhealthy. And there's a fair amount of wiggle. Then during recovery, during the hospital, things are really unstable, which is what we're seeing. And then once he leaves the hospital, there's the blue on the outside, things are starting to be a little less wiggly. And then several months later, you can see that it's become extremely regular, extremely even. And so again, without necessarily understanding the math underneath it, the patient and the clinician can look at this kind of dashboard and gain an intuition for how a thing's progressing. I want to just give one more example about behavioral modification, this time outside the clinic. This was looking at timed feeding. So protocols that Satya Panda has developed at the Salk that we heard about last talk. So this is a roster plot of when an individual is eating meals over the week, and you can see that they're not very structured. And we compared that then to physiology where they were eating in a more structured way. And so here then, this is a week of structured meals, and you can see they're very compliant. They end with a day of fasting, and I won't talk about that here. They logged their meals using MyFitnessPal, and then we taped on thermal sensors to the trunk for core temperature, to the wrist for distal temperature, and used a continuous glucose monitor to look at glucose rhythms. Individually, of course, we can assess the rhythms, and you've seen examples of that. What I was interested in doing also was looking at the network, because where I started this talk was that you want to stabilize these multiple scales of rhythm, because that's what allows optimal function. It reduces friction as systems all agree what time it is, they all agree what they're supposed to be doing. You feel healthier as a result. And so we built using wavelets, transformations of these data. So there's glucose beforehand, and you can see it's jumping all around. Glucose afterwards, starting to become very rhythmic. And using those wavelets and taking the cross product, you can build what's called wavelet coherence. And you can say not only how rhythmic is each individual output at different time scales, but how well do those rhythms agree across systems? So is your body well synchronized, or is it maybe rhythmic but discoordinated? So here's a little dashboard once again, looking at distal temperature, blood glucose, and axial temperature, and the dot size is the strength of the individual rhythm. And then the blueness and the fatness of the edge is how much agreement there was. And so here we see in the week before the person started being regimented, that is pretty crummy. So at the ultradian time scale, on the inside here, 2 to 3 hour rhythms, there's very little coherence. And even at the circadian time scale, things are not agreeing very well. Once they started doing regimented eating, we see that that gets much stronger. And again, with a dashboard of this sort, you can just glance at this thing without a lot of medical training and say, oh, my behavior is definitely affecting my physiology. And then, of course, you can start to think about how that's affecting the context within the neurons of generating these rhythms. And so here again, increased ultradian rhythmicity, increased circadian rhythmicity, and more importantly, increased agreement across these things. So an example of where you're making the physiology apparent to the individual so that it affects their behavioral choice. This is pattern holds across people, but I'm just showing you one individual here. I want to end the talk just by highlighting the fact that wearable sensors are one way of picking up these proxies, but as our behavior has rhythms in it, we actually read rhythmic information in a lot of things we do, including things like online activity. And so there's a lot of opportunity, I think, not just for accessing populations that can afford a new Apple Watch, but larger populations that might nevertheless leave digital wakes in some way so that they too could benefit from this. So this is an example now looking at students and student rhythms of logging into the central management system on campus. This is from Northeastern Illinois University, which is the most diverse university in the Midwest. It's won that award a number of times. So it covers a wide range of people. And working with my friend Aaron Shermer, who's a professor there, we grabbed all of the login times from the central management system. And we did a couple of maps and we said, in red, on those days when students have classes, their rhythms of logging in match class times pretty well, which is here in gray. Whereas on days when they don't have classes, it looks much more biological. You have this much smoother rhythm of when are people logging in, and then, of course, in both days, an absence during the night, so reflecting when are people asleep pretty well. Taking those blue lines, those non-class corrected lines, you can then build personal profiles over time for every individual student. And so here we're looking at how likely is somebody to be active at a given time of day, and the y-axis is 14,000 individuals. And you can see that people fall into what we think of as chronotypes. So there's larks, early morning people. We made up this term finches to be middle of the day people, and then there's night owls. Now, that alone was already exciting. We could see evidence of circadian rhythms and personal profiles from something so sparse as when are they logging into their campus servers. But more interesting was that we could see that this really let us help make behavioral decisions. So we compared these individuals to their times, their same plots, but on those days when they had classes. And you see that this chronotype diversity gets masked, it goes away, because everybody now, so this is the same people in the same order, but now they all, they have to jump when you say jump because they're taking classes. And so their biology is being masked. And what this let us do was compare individuals to themselves across this type of day, with the hypothesis that the more social jet lag, the more disruption that their rhythm experiences on average as a function of class time, the harder a time they would have. And sure enough, that's true. So here, if we look at larks and owls, we're able to see that people that are chronically late tend to have a lot more social jet lag on average, and they tend at all times of day to underperform compared to larks and finches. So we had a sort of hopeful hypothesis that maybe owls would do better in nighttime classes, larks would do better in morning classes, everybody would even out. But it really doesn't seem to be the case, that these owls have a systematic disadvantage from their chronotype. If we get rid of the chronotypes and we just look on average based on social jet lag amplitude, once again, you can see that there's a pretty strong correlation with the amount of social jet lag a student is receiving on average compared to their class times, and their ability to perform in those classes. And so we're not trying to plan experiments both here and there to compare, and in fact with Seattle as well, to get a north-south axis. How do these rhythms change across the year? If you help people choose classes that better match their chronotype, do they get a performance boost? So again, even with very sparse ambient data, gaining insights into rhythms that can affect behavioral choice. So just some cartoons to summarize at the end here. If you think of yourself as a coupled oscillator model, you have things like hormonal secretion being controlled within the brain. So here I have nodes are different hormones, edges are their coupling. Those hormones affect other hormones in the body, or they affect things like body temperature or maybe hunger. And when one of those nodes becomes dysrhythmic for some reason, behavioral imposition in this cartoon, stomach activity because somebody ate a meal at a funny time, then you may not be able to access gastric activity, you may not be able to access hormones every minute. But if you can access something like temperature or heart rate, you can start to tease out the presence of those perturbations. And you can start to then not only assess health, but give that back to either the user or the clinician to modify the behavior to try to reestablish stability. So this is a cartoon from many years ago working with Christopher Pister's lab at Berkeley in miniaturization of sensors. And the idea was related to some experiments we did. If you can have wearable devices or ambient data that's basically aggregating across people and kicking up to the cloud, you can inform somebody like a clinician, or in the case of COVID, maybe a public health officer, about a lot of people at once. You can recognize patterns, for example, where are fevers cropping up in real time, or is my patient getting healthier with their treatment? You can then interact with the patient to help modify that behavior or that treatment choice. And then you can re-aggregate relatively cheaply and at scale the efficacy of those choices. So with that, I'll say thank you. This work, of course, happens because I have the great fortune of being at UCSD, of having an amazing lab, working with groups like the San Diego Supercomputer Center and the Center for Circadian Biology. Some of the many wonderful places that are here at UCSD that make this kind of work, collaborations at scale, really possible and fun. And I will just stop there and happy to take any questions.
Yes, thank you, Ben. Fascinating talk, and so also connected to everyday living, what's happening with people when they're in real world. Are there any questions? I see Gert's hand up. I have a few, but why don't you go ahead? Oh, there are many. Why don't you go ahead, Gert? Oh, that was applauding. Yes, I see. Okay.
Yes, great work, Ben. Yes.
Right. I need to
Thanks,
still
Gert.
get used to these emojis. I'm showing my age here. Nitish, do you want to go ahead if I pronounce your name correctly?
Thank you for a very interesting presentation. For your night owl and day owl, I'll give you one statistic. I interviewed my students, what is the best time to hold a class? And almost unequivocally, it was around 10 to 11 a.m. So I think that's a compromise. The other one was 4 p.m. Anyway, that's a small sample. My question for you is that I'm curious why you don't have independent variables. So heart rate and heart rate variability don't seem to be independent enough. And I suppose if you had some more metabolic, or neural. So metabolic could be obviously temperature, but otherwise physical activity, and neural could be, say, galvanic skin response, because I know you're doing it with wearable. So it seems I'm a little bit surprised that you're taking dependent variables instead of independent variables.
Yeah, no, great question. Actually writing a review about this, I was working on that yesterday. I guess I have two responses. One is I'm just showing you some snippets here, right? So for example, with the COVID work, we had multimodal data that included heart rate variability, respiration, temperature, activity, sleep things. Different wearable devices have different channels, right? Obviously glucose, galvanic skin response, as you mentioned. And then of course, there's the Center for Wearable Sensors here. And so working with people like Joseph Wang, figuring out what else can we measure and throw on the pile. Totally agree with you. I think there's a lot of exploration to do to figure out the level of independence of these different measures. From a coupled oscillator point of view, you would expect that none of them are truly orthogonal in the sense that they're influencing each other as a system. So I think the question is, how do we identify the amount of information gained per channel so that for a given application, say diabetes or pregnancy or Alzheimer's development, what are the ideal channels that your wearable or your ambient sensor would pick up that would maximize that information relevant to you? And we don't know those things.
So it's just a matter of information, more independent, more random, the more the information. So that was
exactly. And the coupling changes over time as well. So it's not just the case that your heart rate goes up, your heart rate goes down. The strength of the influence of one on the other is also rhythmically modulated. And so there's a lot of network mapping that needs to be done to really know where that information is coming from. So I think your question is spot on.
Great. I see my co-chair Shankar's hand. Yes, go ahead.
Ben, thank you for a really, as always, a good talk. Enjoyed it. I want to tie back a question that ties back to the previous talk as well. In terms of the circadian rhythms and also the other rhythms that you talked about, have you investigated the two ends of the aging spectrum, the developmental age of the spectrum, which is, the first formative years of a child when you have growth of all the organs and tissues and maturation of these tissues, and the other end of the spectrum, which is aging, where you have, I'm not talking about disease, I'm talking about normal aging. Have you looked at the effect of these rhythms or the effect of these two ends of the human physiology in terms of your rhythms? And what can we understand from that?
Yeah, great question. Thank you. Personally, I have done a little bit at both ends. I'm getting more into aging, healthy aging, as you mentioned, as opposed to things like Huntington's or dementia, with Dilip Jeste, who's also part of the wonderful ecosystem at UCSD. So we're putting out together long-term studies on not just people into their 70s, but into their hundreds, and what are these rhythms changing, and how does that correlate with satisfaction and well-being and health and that sort of thing. So I think that in terms of healthy aging, I think the jury's out. In terms of pathology, of course, we know a lot of things about how sleep starts to decohere, and in fact, the physiology network that I showed a cartoon of starts to become less coherent as well with aging, and the more that happens, the closer to the end of life you seem to be. As far as the beginning of life, a lot of my postdoc work at Berkeley was the developmental relevance of these rhythms, and a lot of that was in animals, of course, because it's easier to breed in jet lag rodents than people. I wouldn't want people to volunteer for that study, frankly. But looking at apps on nap tracking, Johnson & Johnson had an app where people could log when is their baby sleeping. We worked with them also to do some surface control modeling for how does sleep consolidation work. And you see that when babies are born, they have these beautiful ultradian rhythms, so 3 or 4 nap sessions per day that are pretty columnar on an acronym. So similar times every day. And then they start to do these interesting sort of collapsing convergence behaviors as sleep consolidates. And so again, it's one of these areas that we're just getting into, but there's some interesting implications for sort of what is the advice you can give a mother or a father about when to help their kids sleep to try to speed that consolidation. And then of course, whether or not that's a good thing, right? Are people that when you can find the babies that have consolidated more quickly or less quickly, can you correlate that with other measures of cognitive ability, health, that sort of thing? Because these data are new, we don't know most of these things, right? So we're really figuring out what's the way to interrogate these systems so that we can do those learnings.
Wonderful. Do you have a follow-up?
Yeah, I do have a follow-up, but actually slightly different question as well. So one of the things that I'm really curious about, when you perturb your rhythm, let's take circadian rhythm, we can take other rhythms as well. And clearly it reaches, after a while, it restores back and reaches homeostasis again. And maybe the same homeostasis, maybe slightly perturbed homeostasis, but reaches homeostasis. My question to you is, if you have repeated perturbations, do you get hysteresis effects in circadian rhythms, or you have no hysteresis effects in circadian rhythms? What is the threshold for that?
I don't know that I think it's a great question. I don't know that it's well known exactly what the way in which that history is rendered physiologically, you definitely see additive effects. So, one of the positive findings that came out of some very long-term nurse studies from Harvard, because nurses are doing a lot of rotational shift work, was that your risk for pretty much everything goes up the longer you've been on shift work, right? So as these insults stack up, the odds that something breaks in you also stack up. But the positive finding was that the longer you've stopped doing that, the more those rates then go back down. And so there does seem to be, at the broadest strokes, as you suggest, sort of a homeostatic set point that the body is trying to return to. On the other hand, you can fly to France and stably settle on France time. So obviously you can shift that as well. The extent of the plasticity, the extent of sort of long-term or cellular mechanistic instabilities that you've created as a result, we really don't have resolution on that yet. So again, I think it's a really interesting question. It's part of what motivates me to figure out where can we gather these large populations of data such that we can start to look for that kind of pattern.
Great, great.
So I'm wondering if, for example, flight attendants and pilots would be the right kind of people to look at for this hysteresis effects, because that would be more easily observable if you have monitors.
Yeah, and astronauts. Yeah, no, there's a lot of populations that are sort of subject to this. My approach has not been to reach out to those populations that have previously been working on this kind of thing, but to find the populations that haven't been reached as much. So looking at lower SES or women's health or very young children, things like that, where we still don't really know the basic phenomenology even.
Yeah. I know it's about time. I just have one quick question, Ben. Wonderful talk. You mentioned very interestingly the individual differences, right? Why AI may not work, but I wonder if are we going to reach some point with these wearables and multimodal data to sort of identify individual signatures of these rhythms. In a way, when we think about EEG, if we repeatedly do that, we can say who is who. The signature is out there from the spectral analysis. I just wondered if you combine various signals, whether we would have that kind of signature. And further down, can individuals themselves use their sort of the healthy signature and then try to target that and change behavior in a way. Yeah,
that's beautiful, Ezra. That is exactly what my lab is focused on these days, is what I call physiological diversity. But theoretically, everything you said makes total sense to me, so I agree. I didn't mean to imply AI can't work. It's rather that historically there's been this failure to generalize, that things are well trained on one test population and then they don't work in another population. And in medicine, that often means there's actually negative impacts on groups that already have medical disadvantage, right? So African-American populations, for example, AI is trained in Silicon Valley tend to actually do more harm than good when deployed in these other populations. I think you're exactly right. I think the question is, how do we capture the physiological dynamics that identify somebody, their chronotype, their weekly rhythm, their range, and how do you then also find the sweet spots, right? So maybe sort of like genotypes, you could have your entire sequence, but if you know the right handful of SNPs, you can put yourself in a useful cluster for whether you'll respond to a drug treatment, for example. I think that same kind of information consolidation is probably possible here, where we could identify individuals, but we could also start to identify physiotypes that would be useful, but a lot cheaper to process, a lot easier to make accessible. So yeah, I mean, we'll find out.
Great. Thank you, Ben.
Thank you.
I am thinking we're on time, right? Should we? Shankar, do you want to take?
Yeah, we are quite on time, and Ezra, between you and I, we should lead the discussion, open discussion with speakers and attendees on its transformative clinical advances resulting from synergies between engineering and medicine, including electroceuticals and neurofeedback. And given clearly that you're the clinical leader for this, it'll be fantastic for you to lead off. And I have a couple of points to make, and then we can engage in discussions with our erudite group. And Already I see my, absolutely wonderful colleague, Gert Kovenberg's raising his hand. Maybe we can get him to lead off. Ezra, I will turn it over to you for a moment.
Sure. Yeah. I guess, Gert, you can go ahead if you want. Yes. Just to open up for discussion. Yeah.
Yes, that's one more question for Ben, but this will time quite beautifully in this general discussion, right?
Right.
So you showed us this beautiful data. In fact, it's amazing how much you can tell from just looking at something as simple as temperature or heart rate, right? And so a question, and this is as much as for you as for because all the participants and the entire community here, right? So how can you effectively and directly get brain data in all this, right? Because this is all, in a way, is correlated data. Of course, everything correlates with the brain because all our everything we do is somehow related by our brain states. But so those data, whether it's temperature or heart rate, they're only indirect indications of what is going on in our bodies, right? And so this could be as much as a challenge to the community, so what is your take on how can we extend all that and for measuring more direct activity in the brain, and to what extent is it important? Maybe it isn't.
Oh no, I think it's super important, right? We really want to understand these regulatory networks much better resolution than we do, and as you said, I'm really trying to find proxies and sort of scrape out, claw back the information that I can, but it's noisy. You're definitely more of an expert than I am in terms of the direct neural interfacing. I've done a little ECOG work, but nobody wants to have an ECOG implanted on them. But I think, as people develop smaller, more reliable wearable EEGs, that kind of thing is clearly in the market now and becoming more reliable. I don't really know what the right solution is, right? Those are very shallow. I'm personally more interested in hypothalamic and thalamic relays than cortical activity. And so how do you get that kind of deep imaging safely, continuously? I think that's a big challenge.
Right. So, Ben, you also mentioned during your talk that that not everybody has access to wearables, but we could also potentially look at the patterns at the population level for various things. That was very, very interesting, I think, to see those kind of rhythmicity in those different sort of populations. What would be your take on, for example, what will be the implications of more work to be done in those areas? What can we help the population health if we were to detect the rhythm and intervene? So sort of what will be the implications, I guess?
I think the implications are pretty big as far as I feel. Right. I think Flint, Michigan wouldn't be a surprise 10 years later if you were able to see in real time people's physiology were becoming dysregulated. You could have known something was going wrong.
Right.
You wouldn't have to wait 4 weeks for the CDC to accumulate hospital records to know that people are getting sick and becoming less active, or we're engaged in a research project right now with Intel looking at computer usage stats and changes from pre-COVID to post-COVID. And so you can say, hey, this person's up at 3 a.m. watching Netflix. Normally they don't do that. Like, clearly something has changed, right?
Right.
So I think there's really big issues of equity of access, of security, of all of the ethical concerns about who gets to see what kind of resolution.
Right.
But right now we have basically no lens on most people, and so they are just hoping somebody notices them one day, and mostly we don't. So I think the upshot is enormous, honestly. It's just that it's very challenging to know the right way to do it.
Right. All right, I'm trying to look at the chat to see, are there any questions from the group? Please feel free to raise your hand or go ahead.
I want to just throw your question back at you, because I know you worked much more in this field than me. What do you see coming for being able to monitor actual neural activity in free living humans?
Yes, it's a really great challenge. But it's perhaps surprising that in some places where you wouldn't expect to get brain signals, you can actually get signals, including in the ear. So, so the latest advances, and so there's been great work done in Denmark. We're also working in this area here. There has been demonstrations of in-ear EEG measurements. So basically, those, you can think of those as smart hearing aids or smart ear pods, whichever way you want to call them. And in a way, whether you're a teenager, typically wearing one of those things in their ears, or you're, I guess, more elderly, right? And then you need hearing aids. So typically you have something, or you don't mind having something in your ears. And it's a great place to actually place electrodes. The signals are not quite as great as you would get from scalp EEG recordings, but it's maybe you're down by 15 or so dB of SNR, signal-to-noise ratio. And that translates basically to the decision making. It would be a bit slower. So rather than say telling or discriminating brain states on the order of say hundreds of milliseconds, now you have to wait seconds, and that's okay for many scenarios. And especially when you're looking at the circadian rhythms, then you can observe things over time scales of days. So definitely you can get some reasonable SNR for telling brain states, or at least general brain states.
Okay. I think, Shankar, you have a few slides to present to open up the discussion.
Thank you, Ezra. So I wanted to help lead off the discussion based on something which we did in 2020, January of 2020, before COVID hit us. We had a grand challenges workshop at the interface between medicine and engineering, and this erudite group of 50 people who came to San Diego from all the leading institutions in the country and even some from abroad. We came up with a sort of an outline of a report on what would be the grand challenges. And one of them was the one that I want to present, which ties in very much with the theme of discussion that we have now. And that will give us an idea about what we can, discuss. I hope you can see my presentation. Just 2 slides. And the real grand challenge was making the brain tick. That was identified as the grand challenge. And there are 3 challenges associated with it. The first one was neuro-augmentation of brain function, of human brain function. The challenge is to understand the design principles of the brain and its circuitry and engineer biologically realistic organoids, just like what Rusty was talking about today, that will mimic one or more aspects of brain function and enable deciphering and treating brain pathologies. Essentially, it is to see if we can kind of mimic subsections of the brain that would capture or, that would completely encapsulate what we see in real brain function. So this is, in fact, one of the engineering challenges. It's no longer a biological challenge, it's an engineering challenge, because while we can get transform stem cells into neurons and microglia and astrocytes, how do we get them together into an organoid which should be a functional organoid is an engineering question. And so to that, we need to understand design principles, which requires instrumentation, which requires various ways of looking at the brain at multi-scale. The second challenge was engineering the exocortex, which is the grand challenge is the engineering of human exocortex, which will enable ex vivo measurement of sensory inputs into the human brain and serve as a personalized avatar of the human brain. Essentially, the goal is if I can take my own brain, or one of us can take our own brains, and create a virtual instantiation of this brain so that you can provide inputs into that like we do in normal case, and then see how these inputs translate in this avatar of our brains into an output that we can realistically understand and predict a priori. It's forecasting models of the human brain in a personalized level. And the third one is the neuroscience in everyday world, which is, can we design wearable sensors that are not cumbersome to track human behavior in the context of society, which is essentially not just simply things like iWatch and Fitbit, which will measure all of our rhythms of various kinds, our blood pressure and so on and so forth, but also look at the brain and its function in myriad ways. It can be designed such wearable sensors. So these were the 3 challenges identified as being the hallmark of what would be useful for the engineering approach towards the brain. And and what is needed for doing this, and this group of erudite people identified the following as a requirement. I will be very brief. I just want to say the solution needs are non-invasive technologies for early detection, human brain model systems, including induced neurons and things like what Rusty talked about, vascularized brain organoids, minimally invasive diagnostic markers, systems disease models, developing humanoid models of the brain, replicating brain function, development of control systems for proposed intervention. So it's essentially a control engineering, sensors and controls, measuring the brain to infer brain states, obtaining functional cytoarchitecture of the brain. So these were all regarded as the solution needs. And then enabling technologies include BCI devices, dynamic monitoring and diagnostics, types of things that Ben talked about and things we heard yesterday in our talks, stem cell technologies, brain cytoarchitecture mapping, clinical neurology that ESRA is pioneering, neuroimaging, neurosensing, neuromodulation. So these are all the enabling technologies. We need a lot of people at the core competencies at the interface between people like Ben who can talk multiple languages, neuroscience, AI, computational neuroscience, stem cell biology, etc. And so we need these core competencies. We need partnerships between clinical neurologists, neuroscientists, neuroengineers, systems biologists, etc. And so this is kind of identified as being being what will enable this to happen. But the key challenges are these challenges. So I want to leave leave all of us with this set of questions so we can perhaps focus our discussion on this and also expand to other issues that all of you think are important. So Esra, I hope I have laid kind of the framework for us to have a discussion. And
Sure. Thank you. Yes, thank you for really pointing out to those areas. And there are lots of possibilities. It looks like, it's a broad sort of possibilities, both from the engineering perspective and the clinical perspective. I just don't want to miss, there was a comment earlier before we move on to discussion from John Bunnell. I don't know if the attendees can turn on their microphone and ask their question, Gert. So otherwise I will, yes, they can. John, are you there? Do you want to ask your question or? Okay, so maybe I should read it from the chat. So there is, I think this was mostly directed to Ben's talk. There is this project that some of you may be familiar, all of us, national study. I don't know if Ben, you're familiar. Would it be helpful in a way? The question was large scale studies. Some of the folks in the all of us cohort have wearables. Would it be helpful to sort of leverage that large scale data for your work, in your work? You're muted, I think.
Yeah, no, I've got a couple of graduate students that are specifically interested in this crossover of the wearable signals with deeper omics data, right? So whether it's microbiome or genomics or metabolomics, whatever. Again, where do you find the co-information or the predictive value as you cross these things? I think phenotype, genotype is the sort of keyword thing that gets kicked around. There's more of these things being built, which to me is really exciting. There's a group in Bristol led by Dr. Nick Timpson, who is also doing a similar, many thousands of people across 10 years, sort of midlife. What does British life look like? Lots of different resolutions, including wearables. This kind of study is great, right? I mean, I think it's one of these sort of historic pivot points where instead of spending all of our time generating data, we get to spend more and more of our time analyzing data, and that's super exciting.
Sure. Okay, so going back to Shankar's points, I guess this is an open question to all the engineers who's part of the discussion. Are there any so far existing sort of models, realistic models, biologically realistic models of parts of the brain that are engineered to test some hypothesis or aim to treat diseases? Are there such integrated models of portions of the brain engineered? Can anyone sort of talk about that? Like, for example, I can think of clinically for eyesight, right? Or it's the pathways of vision, you know, could be potentially mimicked in using nanotechnology and engineering. What will be your take on Gert, for example, since nobody's raising their hand? or Benjamin? Yes, there you go. Or Gert, both. Yeah.
Go ahead, Kurt.
Well, Giacomo Indever gave a great talk yesterday, right? So addressing this natural intelligence view on neuromorphic engineering as a means for modeling how the brain works. And of course, the premise of all that is, if once we have a reasonable model of how the brain works, in principle, we can validate hypotheses of what goes wrong in disease. But it's quite challenging, and in fact, it's really a multi-scale problem, and so Tersinovsky will also highlight that a bit later in the discussions that will ensue once we have this IEEE brain panel, right, with a multi-level of scales in representing how the brain works. But yeah, so the multi-scale challenge also means that it's difficult to really to precisely model what is going on in the brain, let alone how to model when it goes wrong. But it's definitely important to move forward in that direction. But Ben, so I'd like to know what your perspectives are on this here.
Yeah, my recollection was that there had been some work, I believe, associated to Mark Ellisman's lab here on campus, where using very high resolution, slicing very thin blocks of brain with electron microscopy, reconstructing then the architecture of cortical columns and modeling sort of activity as a function of that connectome. And it's, I guess I'm both very pessimistic and very optimistic, right? Predicting the weather at some point just doesn't work because there's so much complexity. At the same time, as we make sort of approximations of each, what does a cortical column do kind of ish as a processor, can we model a bunch of them? Of course, we'll have, approximations that allow prediction like that. So I agree with you. I think there's multiple spatial scales, there's multiple temporal scales, of course. And then there's these knock-on effects that Shankar alluded to, where as the system changes, it modifies itself. And so how do you understand glial modification as part of synaptic remapping as a function of the fact that you've been running your simulation over it and it's been making decisions? Not a simple task, but very cool.
Yes. So Shankar, relating to the second challenge, what's your thoughts on virtual reality linking to the second challenge you mentioned, sort of getting these sensory inputs, but through those images that creates different sort of neurotransmitters or state in the brain. And would this be, and then we look at the readout. So would this be something that will be in that concept, or it's totally different? Am I missing the point?
Yes, this was, thank you. This was one of the points that was discussed at the workshop summit that we had discussing grand challenges. I mean, clearly, in order for us to create a virtual environment or virtual reality to look at input-output mapping in the brain, we need to understand the engineered function. We need to engineer such a virtual, environment or a virtual, brain. And that's where the challenges are. We don't know yet how exactly to map. If you, for example, let me give you extreme examples. If you take, for instance, a very small region of the brain circuitry, imagine that you want to look at only coupling of a select set of neurons, pyramidal neurons, for example, in a given context. and then you want to use that to you develop a neuronal model. And there you can really build virtual input-output, and you can create a virtual interface which will really show spikes and where firings happen in different regions of the brain in response to senses. But to do it on a scale that would help us mimic, we need to mimic the human brain function. We need to have really multiple scales of hierarchies of being able to put in such virtual environments together. This is really an engineering challenge. We have two challenges: understanding the design principles, building this together in a form that is more than just simply putting individual pieces together. It has got to make sense at a global level. So these are two challenges that we highlighted in this workshop as being, if we're creating such a virtual environment. And I believe, Nitish, you want to add something to that. You were either involved or you're familiar with what we are talking about. And so, I mean, Nitish will have some erudite comments on this, I have no doubt.
Well, no, there's nothing erudite. So let's be, as I said, more random, more orthogonal the ideas, the more information is created. So I'm going to be quite orthogonal to you, Shankar. I think, again, for Smarr's talk, it seems to me that what we are lacking is multiple quality independent measures. And I echo Gert that, for example, in-ear EEG as an example, if it's a sample of brain activity, I mean, it's a narrow sample, it'll help. And of course, in-ear temperature is obviously easy to pick up. So similarly, we have other things like pulse oximetry that is variable and so on. So I think the problem right now we have is that while the variable sensing is all in vogue, there's a lot of reliance on something like Apple Watch with its limitations. And of course, curating the data and such. So this line of work is big data. It is potentially AI because you will be collecting massive samples from everyone. I think we will get same redundant physiological information from heart rate and activity. And I think we need more neurological, cognitive measures in the context of this conference. There are a lot of posters and topics yesterday about emotions, for example. So, that goes into stress. Now, we cannot get stress response to blood tests and such. So what are the other good variables? So this is in line with this conference to bring mind-body together, where we assess cognitive health, mental health through these variables. And, I hope this can be a good topic next time. If I could just add a thing. So in spirit of this conference, we had technology, and then we have big data analysis, and then we had neuroscience. So I think people tune out, right? They compartmentalize in 3 categories. And I think it's important that actually we don't do that. We bring it together because we're going to need variable with data analysis and machine intelligence coupled to neuroscience to arrive at an answer. So Shankar, you talk about multi-scale, and I'm going to talk about integrating across orthogonal scales. So thank you for.
Nitish, we are on the same page. I'm not, this is not contradictory, absolutely.
No, I think in a way that you make a point to go from reduction of a molecule gene, whatever it is, Rusty Gage's work, let's say we get into all of that to whole brain, and in between an organoid, right? And then there is a gene expression across the scales. So that's one kind of scales. The other one is somewhat orthogonal measurements so that we get high level of data independence and entropy with which we can arrive. And again, that is where big data machine intelligence comes in, because if we had a lot of regressionable data, then we don't need all these algorithms. Similarly, if it fits some straightforward model, and then a standard math can take care of it. It is this large data set that don't fit the models is where somehow these deep learning approaches, somehow they with that mine massive data to derive may be valuable.
Thank you for your comment. I think that you're, in my view, right on target to say that for me, it's a privilege to be at this conference.
By the way, can you turn off the screen so that we can see each other?
Oh, I'm sorry.
Right.
My apologies.
The crosstalk between different disciplines, I think is the key, between sort of the clinician, between someone who's working with wearables and engineers and neuroscientists. And this is wonderfully sort of meshed here, which is, I think, is the way to go to advance the science. I certainly agree that the crosstalk is needed. One cannot individually study these systems, right? Brain is related to the body. We spend a whole career trying to show sleep deprivations related to diabetes. So coming from the brain to your glucose and insulin, I think that this is wonderful. These discussions are wonderful. So Ben, you had something to say. I think you raised your hand.
Yeah, I had maybe it's an engineering question. And I apologize because I'll have to leave in about 4 minutes, but thinking about how you do a neurological exam, not that I do these, but to the extent that I know about them, do your eyes do a certain thing? Are you able to carry out a certain activity? Is your face twitching or not? I know that places like Amazon try to mine images from your phone as you're using it to figure out, are you happy while you're using this app or at this restaurant or whatever they're looking at. And I'm curious whether anybody here has tried that sort of thing to say, if you're measuring something neural, can you, mine facial activity or, saccades or this sort of thing to gain some sort of insight into that neurobehavioral connection?
I think we have a disadvantage compared to the companies like Amazon and Google with their massive data access and computing resources, which is what, right? I mean, a facial is full of so many false positives. We have a small sample of a bunch of students. Similarly, so I think where we can make a contribution in academic scientists is the algorithmic approach with which we can give the fundamental tools, as opposed to us going and trying to gather like something like, I don't know, saccade, right? You take that, you mentioned it. I mean, it's going to be massively problematic with all kind of underlying causal mechanism to just human and genetic variability. So only when you make enough scale of that data and have a model where, I mean, saccade models are very, very well developed. Actually, it's one of the best studied motor nervous system. So within that, and then out beyond that model, so we can, as academic scientists, we could do the model of saccade and how the nervous system regulates it. And then the big data kind of goes around and fits different populations. I wish we as academic would have those kind of resources, and I just don't think we're going to have, and it might be a huge labor. Oh, by the way, we do have student labor. So other than that, we don't have any advantage compared to this big company. So let them do it and we do the algorithms.
I like very much your point, Nitish, of not just top down or bottom up, but middle out. Maybe I call it the middle out strategy, which is to integrate across these two approaches. Clearly, it's a long way off before we can ever get to bottom up approaches. I agree with you completely. Top-down approaches could miss some scales in between. So the middle-out strategy would be the long-term strategy for us to gain something in terms of brain function.
All right, wonderful discussion. So any other comments from the audience, attendees, or questions?
Yeah, we would love to hear from you. So if anyone has comments or views or opinions, I mean, I usually find Metin is not here. I think I can't see him. Metin, are you around? If you're around, you would have some comment. Metin always has wonderful comments.
I would certainly really encourage the students, in my opinion, or experience, I should say, over the years at conferences, the best intriguing questions usually come from students. So please either put it in the chat or turn on your microphone and ask anything you think is worth to ask. Yeah.
And in fact, so we'll have our the demonstrations next, right? And so we have very nice highlights of demonstrations coming up soon.
Sure.
And then we have the breakout rooms where we have real life things happening that very much relate to the discussion we just had here, right? Great. About how to go from unobtrusive neurotechnologies in real time, measuring brain body activity and coupling that to clinical outcomes, at least in the long term, but at least initially doing science, but eventually and engineering, but eventually going towards clinical translation.
Great, great. So are we taking a break now? If there are no further questions, I'm just maybe not following the schedule here.
Yeah, I think we should take a brief break, and then coming back, we have, I believe, a short. Gert, correct me if I'm wrong. We have short, quick, glimpses of the poster presentations, and then followed by demonstrations, followed by demonstrations.
Yes, so we have the highlights of the demonstrations coming up in about 9 minutes. And
it'll be chaired by Nitish and somebody else. Is that, I'm sorry,
it's Marianne Chinechi and Nitish Tekor will be leading the
Mariam is the chair. I'm the
highlights.
backup. Okay, so it's Mariam.
Nitish, I never known you as a backup for anything. So therefore, anyway, you're co-chairs for this. So this is great. So with that, I want to thank the audience for the presentation. My very distinguished co-chair, Esra Tesali, and Gert Kallenbergs, of course. And then I think we are done with this session for now. Gert, please correct me if you want to add anything.
Thank you all. Yes.
Thanks, everyone. I'll see you soon. So we have a short break now.
Thank you.
I think we're ready to get started. Marianne or Nitesh?
Yeah, so we can get started. You will be flipping the slides, correct?
Sure, yes, yeah. Thank you. Thanks, Marianne.
So hi, everyone, and welcome to our demonstration highlights for the day. We have 6 beautiful demos, and we're going to go flip through the slides. When each of the demo slide comes up, the presenters can present they have about 3 minutes, 2 minutes, 45 seconds to present. And then we'll move on to the next slide for the next demo presentations. And then after a 15-minute highlight, we'll go to breakout rooms. There'll be 2 breakout rooms, 3 demos in each breakout room that you're welcome to attend, where they can expand on their demos there. And with that, I think we can start going to the first demonstration.
Okay, that's us. Can you hear me?
Yes, we can.
Okay, perfect. Then hi, glad to introduce myself. My name is Laura. I'm from Mentalab. We're a Munich startup focusing on mobile biosignal data acquisition. On the screen there, you see me on the right wearing our new technology that's called Mentalab Explore. What we have is a wireless EXG device. So that is a device that can record multiple types of biosignal data, such as EEG, EMG, ECG, and so on. We also have movement sensors, and what we'll be doing in this demo is, first of all, to give you an overview of what kind of technology we've got, of its key factors, some of them being the very compact size, as you can see on the picture, its research-grade data quality that might be interesting to some of you, its flexibility that is unique compared to other providers, and above all, also its open source character regarding the software. In the demo, I'll give a short intro to who we are and how this technology has come about. I'll give you some key information about the Explore system that might be relevant, above all, for the technologically minded and research interested among you. We'll be including the open source software options, and I'll tell you more about that. And yeah, I'll give you an overview for what kind of areas this technology is relevant. Maybe your area is too. After giving this little background, we'll move on to the live part of the demo, where I will be streaming EEG data from my brain live directly to you. You'll see my screen and you'll get some insight into my brain in live time, for so to say. And with that, I hope you can judge the technology and its quality for yourself. I will keep the boring descriptive part short. I will show you whatever you want to see, and then we will open up for questions. We have many materials to show, including other demo videos and other applications. We can go into more detail. I will adapt that according to whoever is present and according to your interests. So I hope you will join us on this discussion about the new wave of wireless wearable technology and research. So I hope to see you there and looking forward to it. Thank you very much.
Thank you. So next, you can start.
Sure. Hi, my name is actually Paul from UC San Diego, works in the lab, Dr. Gert Kallenberg. So my demo will focus on this new chip that we've developed, which we call NISOC, stands for the Neural Interface System on Chip. It's a 32-channel bioamplifier that allows for low power, low noise, simultaneous recording of 32 channels. The intended application, as you're seeing in the video, ranges anything from direct recording from neurons. So in this clip, this is a playback from leech neurons, action potentials. So one of the major highlights of this chip is not only that it's low power and that it's 1 by 1 millimeter, but that it also allows both voltage clamping and current clamping capabilities, which we'll get into further details during the demo as to why that can be important for various applications in neuroscience. So things ranging from deep brain stimulation and recording to cortical surface recording, LFPs, obviously EEG, as well as more broader body and brain health sensing, things such as electrodermal activity, impedance scanning, and just overall E-star-G. Yeah, some of the other highlights of the chip is, it can sample right now up to 25 kilohertz. We're measuring around microwatt per channel of power consumption. Yeah, and I'm looking forward to showing you our demo and talking further about the potential applications of this chip.
Thank you very much. We can go to the next slide. Hello, go ahead.
Hello, my name is Samal, and today I'm going to demonstrate some neuromodulatory principles in silicon using Neuroden, a digitally programmable continuous time analog neuromorphic chip, which models neuronal membrane voltage dynamics and ion channel kinetics of generalized Hodgkin-Huxley neurons with conductance-based synapses. So one of the fundamental questions being answered by neuromodulatory control is how biological neurons can achieve robust and reliable activity patterns despite their high degree of uncertainty and variability in their individual components, i.e. the neurons and synapses. So here we'll show that by modulating the maximal conductance parameters of, say, typical conductances found in a Hodgkin-Huxley neurons like sodium and potassium. So by modulating the maximal conductance and the opening and closing rates of the ion channels, transitions between different behaviors such as spiking and non-spiking can be seen, as well as properties of the waveform such as frequency of spikes can be modulated. Finally, we'll also demonstrate antiphase oscillations between a pair of tonic spiking neurons interconnected by inhibitory synapses by modulation of the synaptic conductances. Thank you.
Thank you very much. Next slide. That will be in breakout room 2 later.
Okay, so it's me again. So again, this is Akshay Paul from Dr. Kallenberg's lab here at UC San Diego. So our other demo today is going to focus on this unobtrusive wearable health sensing platform that we are one of the few labs in the world that's kind of pioneering, and that's for in-ear EEG. So as you can see here in the image, it's not just limited to the EEG. So we can get other modalities of measurements as well. So starting with EEG, measurements taken completely inside the ear allow us to get auditory evoked potentials, electrodermal activity, auditory steady state responses, as well as the impedance scanning of the brain. More recently, we've seen that we can detect alpha modulation of the brain from completely inside the ear. We can also get, to some degree, eye movements. So EOG, we can get blinks. From inside the ear canal, we can pick up EMG and electrodermal activity. So in the image, we're seeing that there are also electrodes placed on the forehead in the conventional 10-20 forehead positions, and then also under the eye for EOG and EMG. So those are for comparison for what we see inside the ear. And for the most part, they're quite comparable signals that we get inside the ear are correlated to the conventional positions. So the data acquisition unit that you're seeing placed behind the ear, that's fully contained, completely wireless, a 16-channel bioamplifier that we call the WEDAC, so the wireless electrophysiology data acquisition unit. As I mentioned, it provides 16 channels of simultaneous recording up to 4 kilohertz. The wireless protocol is a Wi-Fi protocol. It allows many of these devices actually to be placed on the body, each providing 16 channels for different multimodal sensing, all on the same network. And then the data is streamed to live streaming layer, for example, or it can be sent to AWS cloud for cloud computing and visualization on a smartphone or on a PC. So, so for this one, we have a pretty robust pre-recorded demo that we're looking forward to showing you and answering any questions that you might have.
Thank you. Thank you. Next, next demo.
Hi, this is Yaling Huang from National Yangming Jiao Tong University at Taiwan. I want to share Expressible, an open source software for decoding EEG using novel AI models with user-friendly GUI without any programming. Expressible provides a fast, simplified, and user-friendly solution of EEG decoding for investigators across disciplines to leverage cutting-edge methods in brain neuroscience research. Available functions include model training, evaluation, and parameter visualization in terms of spatial and temporal representations. The left figure is the main dashboard that displays the status of the modeling pipeline with information about data loading, model trend setting, and model status. And the middle figure is the underlying spatial patterns and visualized with the topography of scope, which is a straightforward tool to convey the spatial distribution of kernel weight. And the temporal convolutional kernels can be visualized by waveform observation and spectral analysis to characterize in time and frequency domain. These visualization techniques adequately transform complex CN models from black boxes into glass boxes, providing understandable and explainable representation that are compatible to conventional insight of neuroscience. If you'd like to decode your EEG data with Expressible, please make sure to visit our live demo. Thanks for your attention.
Thank you. Next, we have next presenter, Salah Hamdi. You there?
Hello.
Hello. Yeah, hi, we hear you.
My name is Salah Hamdi. I am from Tunisia. I'm Doctor of Computer Science in the Faculty of Medicine of Monastir. So my paper is entitled Autoregressive Causal Statistical Inference from Online EEG and ECG Signals. So we'll study of the interdependence between EEG and ECG physical signals. We'll use of time series and autoregressive vector called VAR for modeling both ECG and the signals. And we'll look for causality between those signals. We'll look for causality between both those signals, ECG and EEG. So the objective is to look for correlation between those signals and the study heart rate synchronization. And we'll apply on normal patient. So we'll demonstrate our granular causality. The reality exists between ECG and EEG signals. Okay, that's all. Thank you.
Thank you. I believe there's one more. Is there? Or that's it, I think. We had 6 demos. I think we went through all of them. Next, we will have two breakout rooms. The first three demos will be presented in the first breakout room, the last three demos in the second breakout room, and we look forward to further discussions and elaborations there.
Thank you, everyone. So now we'll be opening the breakout room. So please go and join those rooms. If you have any trouble joining those breakout rooms, make sure you have the latest version of Zoom 5.3 or later. Otherwise, you may have trouble actually moving around. If you have any trouble, just let us know. We can help. And it seems to be working. We really have 22 joining the multimodal neural interfaces.
There's nobody joining the other one though. Is that?
They probably start with number one. That's the.
I'm
now
sorry.
we have 2 or 3 in the.
Okay, now there's some people there. Okay.
I guess everyone just wants to go to the first one first, right?
I see. Okay, maybe I'll go to the second one first then. Join them.
Great idea.
Hi, I was looking for that breakout room with some sensor. I don't find it yet here. There's some kind of sensor.
There's multimodal neural interfaces or real-time brain state decoding. Which one would you like to be in?
Oh, okay. Oh, no, multimodal, yeah, that one.
Okay, I'll move you there.
Thanks.
So welcome back, everyone, from the great demonstrations that we had. So we're very delighted to have Amy Cruz, who will be leading a panel session with industry and clinicians. So very much looking forward. Amy, yes.
Excellent. Thank you so much. Thanks for the opportunity to share the ideas in this panel and to lead it in this, quite exciting topic. I think we're taking some things to some new places, and so would like to sort of call up to the stage, as it were. Hopefully, you can see the slide. Can you see that, Gert?
Yeah, we can see it. Perfect.
Okay, perfect. Excellent. Call to the stage, as it were. Jose contacted me yesterday and said he might not be able to join. So if he is, he's joining us from the airport. So we have Jose Pons, Ellis Mang, Tim Mullen, Miriam Shaneshi, and Ezra Talsali joining us today. The idea behind the panel is that each of the panelists would share some kind of opening thoughts up front on this sort of critical topic in terms of transforming innovation in sustainable technology. And those of us who've been working in this space for a while are very curious, I think, about how these technologies will not only scale, but will actually be able to reach the maximum number of people, individuals, and patients as possible. And so I think these are some of the overarching questions that we have in this theme. And so I guess I would just to check and see if Jose is on the line or not. Jose, are you here? I didn't think so. So then Ellis, I was figuring maybe we'd go to your opening statements, and I have your slides, so I will advance for you there if you want to open it up, Ellis.
Sure. Thanks, Amy, for the introduction, and thanks to the organizers for putting this on. So I wanted to kind of pick up this conversation. I'm hoping that we can be interactive today by picking up off of where we had some conversations yesterday. I think there was some discussion earlier in the morning. How do you get access to some of the stuff that you're seeing, whether it's chips or electrodes or anything else? And so what I try to do here is maybe motivate this and also share with you one idea of how to actually share some of this stuff and to get access to it. And also highlight some of the challenges, because I think there are many, and I think it'll take the whole community to have an active conversation to figure out how to fix these things or to find paths forward. So if we will look at technology, so I myself develop devices, and so I'm looking at this from very much a technology developer, researcher point of view. And so within this space, you might very loosely group available technologies such as electrodes, circuits, integrated systems. I know there's software out there. I don't really do software, so sorry, I'm leaving that out. And if you look at the mode in which the research is done in these fields, we look and we see that there's varying levels of technology readiness and also availability of facilities, talent, and other resources to support that. So you heard a little bit about some neural interface devices. So I'm kind of just calling neural interfaces very simply as electrodes. And typically the way that research is done is that they're designed and fabricated by researchers. And there's limited access to these technologies by anyone else who's not in the business of doing this device design. And circuits, there, I think the infrastructure is a little bit different in the sense that the integrated circuit world has been around for quite some time, and the technologies to support building those is quite mature. And so you don't have to have access to a foundry within a university. In fact, very few people build circuits in a university setting right now. Most are design the circuits and then send them out to get fabricated by professional foundries. And there's a number of different ones available and methods to do this. And so this is sort of a well-worn path by many. When it gets to the point where you're trying to put electrodes and circuits together into systems that can then, say, be implanted or used on the body, typically researchers have to fill in the gap and bridge the two worlds of electrodes and circuits. A little bit is done by the electrode researchers, a little bit is done by the circuit researchers. And then there's sort of some of these that actually will go to the step of doing integration. And it's not sometimes always straightforward because you don't have control of all aspects of that. So you're at the mercy of what exists out there. And so if we want to look at some of the gaps, I've talked a little bit about some of them earlier. We're realistically, for the research world, talking about devices that are produced in low volumes. And if you want access to something and you don't have the talent locally to do that or the right facilities, you're really at the mercy of what's available. And there's really not a lot commercially. Many researchers want something custom, and that's a challenge, right? Because of the low volumes. There are now funded dissemination efforts by many groups enabled by some of the funding in the BRAIN Initiative and also other efforts around the world. The challenge there is that you're limited to the designs that have already been developed. Typically, those designs are available in fixed numbers, and so you don't have access to lots of devices if you're planning on doing research with that for some period of time. And frequently the research solutions that are developed require a great deal of support to be able to bring somebody else up to be able to implement those. And so, having an ecosystem that helps with education and training and hands-on training, which is difficult nowadays still, and providing the right documentation is tough, and having a support network of other users who you can learn from, that's not necessarily widely available. Many of the dissemination efforts are limited in terms of time, in terms of scope. And so I would say now it's not truly sustainable. And it's not necessarily something where you can just push it off in the industry. A lot of times you hear researchers say, well, that's not something that we do in research. Industry can do that. I'm not sure that that's the solution, because the sort of heavy lift as far as support that's required in low volumes are not compatible with an industry model where they actually need income to be able to survive. So it's not clear to me that this is a solution that's viable moving forward. So we need conversations in the community to figure this out. So is there something that we can do in the interim? And so, at the very end, I have the segue to the next slide, which is a solution, a potential solution. It's not clear that it itself is sustainable, but can we leverage, at least in the world of electrodes, the infrastructure that exists in the integrated circuit world, or sort of the well-worn path that they took to be able to make integrated circuits more widely accessible without having to have that specific facility local to you. And so with support from the National Institutes of Health, we now are hosting a resource center that is based on the model used in the integrated circuit world, where we have the ability to produce custom devices for researchers on demand. The service is free currently for use as we start to figure out if there is a sustainability path moving forward. And we also have, for those who don't want to go and invest the time and effort into doing their own custom design and are happy to work with something off the shelf, those as well. And so yesterday there was a poster on this. If you didn't catch that, some of the pictures are below, and you can find more information on the website or contact us if you're interested in getting access to this resource. I'll note that although I'm focusing more on the devices here, I have to acknowledge that the devices by themselves really need support beyond a manual, but also how to handle them, how to implement them surgically, and in some cases also how to implement them in neuroscience experiments. And so we do have some of that support. It's difficult, I think, to carry out given COVID, but to date, we've been in operation maybe about 2 years or so. We're in our 3rd year. We've shipped hundreds of devices in that time all over the world. So I'll leave it at that and turn it back to Amy.
Thank you, Alice. That's actually very that's exciting. I wasn't aware of that capability, and maybe some others weren't as well. So I'm excited to hear what people's thoughts are about that. I'd like to call to the virtual stage now my friend and colleague Tim Mullen from Intheon, who will hopefully share with us his thoughts on this topic in his opening remarks. Thanks, Tim.
Hi, great to be here. Can you hear me okay? Am I coming through, Amy? Just want to make sure my speaker's working.
You're good.
Oh, wonderful. So yeah, to resonate on this point about access, this is I think a really important, very important question for how we transform global health through innovation in a sustainable way, in a cost-effective way. I like to think about Geoffrey Moore's famous dialogue on crossing the chasm between product ideation and mainstream adoption. And in the neurotech domain, we have a similar chasm between the basic science and research and development and the industry and clinical applications that ultimately drive user adoption, market growth, and sustainable businesses. And on the one side of the basic science and research, we've got these labs that have all this great expertise in basic and applied research, experimental design, algorithm development, et cetera. A lot of money has gone into these labs over many, many decades, creating really an immense amount of capital in the academic institution that can be leveraged to try to create new breakthrough therapies. On the other side of the equation, we have these industry teams that have great expertise in things like product ideation and design and scalable engineering, marketing, et cetera. But there's really a fundamental difference in the way that these two different teams historically have operated. On the academic side, development progresses relatively methodically in kind of constrained lab settings. It's intended to yield sort of maximum scientific knowledge. And then on the industry side, it's primarily an issue of getting product into market as quickly as possible, ensuring they perform suitably well for their intended application, not perfectly, but well enough, and often in sort of complex, unpredictable environments. There's sometimes different goals there in terms of what each group is trying to achieve and their ability to coordinate around a common outcome or even translate anything from academia into real world practice. So, one thing that I've seen over now, 15 years looking at this space is that to increase dissemination, we really need to not just cross this chasm between these two different groups, between the academic ideation and incubation and the application, but really close it, bring the two worlds together so that product teams can easily build on the state of the art in scientific R&D and iterate quickly, and that labs can conduct research in real world context, which reduces the translational cost. If I've innovated heavily in a lab, identified maybe some great neurotech application that works great in a lab, but then as soon as I translate it into the real world, I find it actually fails because I failed to consider a lot of contextual factors. That creates an immediate barrier to my dissemination, and immediately the cost to translation goes up dramatically. If I had conversely worked closely with industry partners, perhaps at the time even of developing the research, to ensure that I'm understanding how this works in the context in which the product will ultimately be realized, now we may be able to significantly reduce that barrier and increase that access to all the know-how and capabilities the labs can offer. So an important thing here is just to understand how value is created for each group, and importantly recognize that entrepreneurs have different pressures than scientists. So like a lab might need to get high impact research out, startup needs to get to market as quickly as possible, as I mentioned. Those are important things. And then I like to think about really 6 main categories of effort that can increase this dissemination, which again, all leads to cost reduction and to sort of sustainable businesses in this space. With access, we talk about accessibility, clinical utility, consumer utility, ethical frameworks and how those work and how those ensure they're serving the people that they're intended for, and standardization and sales. And basically the earmarks I wanted to put here that we can talk about points on later is with accessibility, it's really making possible access to industry-oriented hardware, software, and data platforms that industry partners can leverage that are at the same quality and caliber that a research lab would be used to using. So for instance, if I'm a company that wants to develop a new neurotherapeutic, I would like to be able to avail myself of hardware or software that is readily accessible, that is cost effective, and that is at the same caliber as MIT, Berkeley, Cornell, whatever university Stanford is using. So that's point number 1 with accessibility. 2, clinical utility, just really thinking carefully about the patient's needs and their desires and ensuring that we have really streamlined, clear regulatory processes that can adapt to the pace of emerging technology, and ensuring again that those regulatory frameworks are designed around both academic concerns and industry concerns so that there's a faster path through the regulatory process from that initial ideation, coming up with that new biomarker, that new therapy, to productization. And then consumer utility is really just identifying killer apps, thinking carefully about the ergonomics, the user experience design as early as possible. The ethics is really all about ensuring that at the very beginning of you building your technology, you're building the ethics in. So working with academic neuroethicists early on, so that you're not finding out halfway into the product development cycle that, oh, wait, we made some really, maybe some not so good decisions here that is going to lead to maybe a use of our product that was not so well intended. And now you got to go and recalibrate and rethink that. Again, increases expense, increases a barrier to dissemination. And then finally, standards are essential to reducing cost. Having good standards that allow for interoperability between hardware, software, data, and between particularly what academics are using and what industry partners are using. So like if I am using a piece of hardware in my company, I would like to be able to interoperate that piece of hardware with hardware that a lab is using. I would also like to ensure that data that a lab is producing is something that I, as an industry partner, can easily use, can leverage, can identify a biomarker in. So, or maybe even combine my clinical trial data with a lot of academic data that's being produced to maybe find something new and interesting. This requires standardization. And then the last thing is just the sales and marketing part. One thing that I've seen a lot of is that there's a great deal of early, or there's a great deal of ideation that happens in the academic environment that doesn't really involve anybody from a sales side of the equation until much later on. Maybe it's like, okay, now I've spun my company out and I now do a bunch of stuff, and then I hire a marketing person, and then they say, you know what you think the user wants is really actually not at all what they want. So a lot of the value that can come from bringing marketing and salespeople in very early, even in the early R&D stages, is that it can help inform an understanding of, is the thing that we're innovating for actually something that the end user will adopt, will use, and be useful to them, be interesting to them, be essentially productizable. So I do think that that's just something that has to come into earlier in the conversation, and maybe even at the stage of labs who are thinking about translating their technology out, to bring those kinds of people in early. So those are a few things that I think, a few perspectives. I know it's a lot of different prongs, but if you
no, it's actually great. There's a lot to talk about. There's a lot. Okay. There's a lot to work with.
I'll hand the baton.
Yes, thank you. Thank you, Tim. It's going to be lively later, I know. Well, Miriam, Shaneshi, would you like to take the virtual stage and share some of your thoughts with us in this opening statement?
Sure. So actually, I think I can cover the software part of things, and Ellis and Tim covered part of the hardware issues that are at play. So I'm mainly an algorithm designer, develop data science and machine learning methods to discover biomarkers and build algorithmic technologies around the brain. And I would say, from the point of view of software and algorithm design, one challenge that I see is that we develop these methods in our labs, and we show that they perform well. They can find these neurobiomarkers and do stimulation and neuromodulation in some way. But what we do not take into account are the device limitations that that manufacturers have. For example, whether it's Medtronic and with their neuromodulation device, what kind of neural features we can extract, what kind of computations we can actually implement in their device. So a lot of our labs kind of develop these algorithms, and then they may not even be implementable in current medical devices. So one issue that I've seen for the wide dissemination of some of these methods is that they may not be applicable to today's devices. Of course, there's a lot of beautiful work done on improving and advancing medical hardware and devices. But before that happens, I feel like some closer collaborations between industry and labs, scientific labs, who are building also machine learning methods to figure out, okay, once we have the data and once we have these hardware capabilities, what do we do with it, would be quite important in having these systems clinically viable and available to a wider population of patients.
Excellent. Thank you, Miriam. That's I'm glad you brought up the software side of the equation, and I think we're hearing some themes here. So for my last panelist today, unless Jose jumped on before he got on this flight, is Esra Tassali. If you wouldn't mind joining us on the virtual stage and sharing some of your opening thoughts with us. Keep clicking. Sorry about that. There you go, Esra.
Thank you, everyone. This is wonderful symposium, and I'm pleased to be part of it. First of all, I must thank all the trainees, students, as well as the faculty who have shown all these very exciting demos and novel technology. When I think about transforming health coming from a clinical perspective, I think of two things. Prevention, right? Disease prevention, and then intervention, treatment. So, and some of these concepts earlier mentioned, I would like to see technology being developed, patient-centered, user-centered, right? And also really we need to identify the clinical gaps, clinical needs, and fill them with the advanced technology. The other part that was part of the discussions today earlier at the symposium is the crosstalk between engineering team, between biomedical faculty, and most importantly, the physicians, clinicians, and the patients themselves who would be using the technology or taking advantage of the technology. And then finally, I think I would also like to put out this concept of sustainable technology, right? Like you may come up with something new, form a company, maybe in fashion for a while, but you have to always think longer term. What are my next steps? How this could be expanded to different kind of technologies, evolving very fast. To still continue to help our patients or even healthy individuals in terms of prevention perspective. So I would leave it brief and hope to hear from the audience what their feedback is. Thank you.
Excellent. Thank you. Thank you, panelists. So now I'd like to, I have a couple questions to start. Gert, I know you probably have a couple questions to start. And then the opening it up, certainly if you want to put open questions or if there's a question in the chat. So maybe I'll just open it up to the panelists to start with, and I'll share a little about myself. I'm a neuroscientist by training, but have made my way through various stages of my career. I was once a program manager at DARPA. I've been in both small and large industry, primarily in defense. Was most recently at a startup trying to do commercial work in applied neuroscience. And now I'm a general partner to a venture capital firm. And so through those different lenses, I've sort of seen the neurotechnology industry and these technologies evolve. And still yet, I think over this time frame, we've heard some of the repeated themes. And so I guess I would like to open it back up to the panel, since it was a topic that came up repeatedly. What are the mechanisms or the best mechanisms that we have either conceived of or not have conceived of in terms of this crosstalk that we keep talking about between academia and industry? And again, certainly from my perspective, I see lots of companies that have just spun out, as it were, in various stages of maturity. Some should have stayed spun in for a little longer. And so, what are the mechanisms that are out there now? What's working, what's not working? Maybe just some thoughts across the panel. I'll open it up broadly, and maybe you can play off each other in terms of the thoughts there.
Maybe I'll start off. So actually, I love the mix of different expertise on this panel. I think it's great. We're also anchored by a funder too that has a very different perspective. A lot of points were brought up, and there are so many different angles I think you can look at where the challenges are and why some of these problems have persisted and have not been solved. I think from the clinical side, if we start where Ezra kind of left off, there are many challenges that don't get communicated to the folks that are working on new technologies. And then, you know, having a part of that challenge is that sometimes what is needed in the clinic doesn't make for a good research topic that can graduate a PhD. And then, you know, how do you get that problem solved? That's a persistent challenge. One way I think that it's kind of worked at USC is we can actually get some of these connections at the undergraduate level into capstone classes where they can work on these projects as part of their education. But the challenge there is, you know, having, I think, enough time to be able to finish up something and then return that back to the clinic. So that's a possibility, but maybe not something that is going to be that impactful down the road, because students come and go. These classes happen within a specific time frame. We have to actually finish and start and finish within the confines of a semester. And so it's not a perfect solution, but it is one solution probably of many that can be come up with. So that's on the clinical side. You know, as far as motivations for bringing the different sides together, I think Tim was saying that what drives industry and what drives academics is very different, and that's always a challenge. We oftentimes have to present something that is new, far out, very shiny and different in order to convince our colleagues in academia to want to rate a proposal highly and have it get funded. But that's sometimes at odds of what's practical. And there are folks that want to do sort of the blue sky research, and others who want to see what they do then get translated and help the clinic. And I think that's something that I don't see a good solution to. Right? How can you change academic merit review and tenure and promotion guidelines to convince people to want to actually create more real world impact? That's a very challenging thing.
I guess I can jump in. One promising aspect that I see over time developing, at least at the NIH level,